Morning, morning. My name is Dr. Diva Amon. I'm the Chief Scientist of Deep Wonders of Trinidad and Tobago expedition that we're on right now. We have just touched down at 1,300 meters depth, just about. And I am really excited for this dive. This dive is very special for me. We have visited this site before. It is one of a handful of locations in Trinidad and Tobago's waters that have been explored by scientists before, in the 1980s by a French team, and then again in 2014 by the EV Nautilus. And what they found at this site is, I'm obviously biased, but is extraordinary. These oases of life in the deep sea, methane seeps that are just laden with many different species and, thousands to millions of each of them. So stay tuned. You're in for a real treat today. We did map it last night, and we did find, again, indications of seepage. These bubble streams, the fact that the seafloor is very hard. We could see the bubble streams in multiple types of data. So we are very certain, barring getting our eyes on it, we are very certain that these seeps are still active. And it'll be really interesting to see how they've potentially changed, and if they've changed since the 2014 visit.
So this first waypoint, right?
Yeah.
Is that where we're putting the off seep?
Yes. So before we get to the seeps, I'm going to hand over to Dr. Anna Metaxas, Professor Anna Metaxas, who is going to guide us as we deploy some experiments. All yours. Okay, I'm going to be right in the What's happened to my pencil?
Well, I'm zoomed out.
We're almost there, right?
We're getting close. Yeah, we had to go a little north because of the current on the way in.
Yeah.
We're working our way down.
So 1309 is
Yeah, that's our
target.
Okay. Putting on down there.
Where's the current coming from? Oh, okay. Let's see. Take us a while.
Yeah, I just had to slow it. First I can take off, but then my vocal
Yes.
starts doing that.
Yeah.
We're probably halfway.
Good morning, everybody. My name is Anna Metaxas. I am a professor at Dalhousie University in Nova Scotia on the east coast of Canada. And I'm here as a guest of Dr. Amon, who's the chief scientist of this cruise, to participate mainly in the ROV dives that we're doing. So today is a very special day, as Dr. Amon already said, for her personally, because this was a place that she visited back in 2014. And it's, in general, a special day because we are going to be visiting methane seeps. And so at this point, you don't see any methane seeps. What you see is the seabed that we've been seeing for the many of the previous footage from the previous dives. And that is because cold methane seeps are actually surrounded, as Deva mentioned, they're essentially oases in the deep sea, and they're surrounded by the sedimentary communities that we typically see. They're more characteristic of these depths. And so what makes methane seeps be oases is the fact that methane is actually seeping through the seabed in bubbles into the bottom of the ocean in the water. And so what that allows animals to do is take advantage of those methane bubbles and create the communities that we see. And the way this happens is, as we've kept mentioning earlier in our previous dives, is deep sea is extremely nutrient depauperate. So it doesn't have a lot of nutrients. And so the way food reaches deep sea organisms is by dead plant and animal material that sink from the sea surface. And so that really is not a lot of material. Typically only 1% of that material will actually land on the seabed at 1,000 meters, or the types of depths that we're diving in, between 1,000 and 2,000 meters. So it's not a lot of food. And so what has happened and what we see in the sediments is not a lot of animals. So we don't see large populations. What we see is diversity. So we see a lot of different types of animals that have developed different adaptations to be able to take advantage of those different types of food and the low amount of food. Now, what happens at methane seeps is they're actually a form of carbon synthesis that happens just like photosynthesis happens at the sea surface by plants. Similar type of synthesis, but it's done by chemistry. So it's called chemosynthesis, happens at those methane seeps. And so there are microbes, bacteria, that as plants do, basically utilize the methane from the bubbles that come out of seep through the seabed and oxygen from surrounding seawater and essentially chemosynthesize. So they produce carbohydrates, and so that's how they grow. It's that they are sort of the plants of the deep sea, except instead of light, what is used for the energy to drive that production of carbon is the energy that is released by the interaction between methane, and it's a chemical reaction. It generates energy. That energy is then used for synthesis of carbohydrates by microbes. And so now we have the equivalent of a plant that has used a chemical that seeps from the deep ocean. So you have local production of food, local carbon production. And so that allows for a lot more animals to be able to grow because the food is not limiting anymore. There's a lot more food because the animals don't rely from what is going to rain from up above. And so we have, so what we will see, very different, what we expect to see is that we'll be going along seeing this type of environment, and then all of a sudden there will be a lot of different animals like mussels and tube worms and other fish and crabs, etc. And so that's why they're called oases, because they have this large biomass that you don't see anywhere else. Now, diversity is lower in those environments than you would expect in this, more sedimented environments that we have been seeing as we're traveling along. And the reason for that is because for animals to be able to take advantage of those particular microbes, they have to have special adaptations. And so it's not a large number of animals that have developed those adaptations, and so there are only some specific organisms that have. And one of the adaptations that they have developed is symbiosis. So you may know that corals in shallow waters have the symbionts that basically do the photosynthesis for the coral, and they produce carbon for the coral. And so it's a similar situation in cold seeps, except now we have the microbes that may live in mussels or in tube worms, and they actually do the same. They're symbionts, and they basically use the methane and the seawater, the oxygen from the seawater, to produce carbon that then is being utilized by the animal they live in. And so they're what are called endosymbionts, so they're found within the animals, within the tube worms, within the mussels. And so as for symbionts in coral reefs, the same thing happens here. As they grow, then the animals that they're living in can actually either eat them, so digest them, or actually use carbohydrates that are released into their bloodstream. And so in order, these are pretty special adaptations. And so, this is why we only have a small number of species that have developed these adaptations and are able to take advantage of the methane released in seeps. So we're still we're almost there. We're almost at a location which is near the seeps, but it's sedimented and it has no evidence of seepage as far as we could see on the mapping that we did. And so what this has been recently termed, or relatively recently, as a peripheral environment habitat, and that means it's on the periphery of the seep. It's still sedimented, as you see, but it's not. There's no evidence seepage, and so the idea is that there's also no influence from the seep itself. So, because water moves through ocean circulation, what happens is food can get transported. You can see particles in the water. When we'll get to the seep, there may be more particles. And so those particles are actually food that then may be used by organisms that live not on the seeps, but adjacent to the seeps. And so that means that there is still some influence of the seep in the surrounding environment. So if we move far enough, a certain distance, and that distance will vary depending on currents, depending on how big the seep is, etc. We should be in an area that the organisms that live here do not feel, if you will, the seep. There's no influence of the seep in this habitat. And so we have almost arrived at a place, at that target location, away from the influence of the seeps. And what we have planned for today is to deploy an experiment. So a lot of the work, most all of the work that we have done this far on this research cruise has been exploration. We have been to places that we haven't seen before, and the first item of business when you do exploration is to try to map the area, both geologically, but also map the animals that live there. And so we've been doing this with video, but then once we have an understanding of what the animals are and what potential interactions between different animals are, we try to run experiments to understand why animals are in particular locations, why particular habitats look like they do. And one of the sort of particularly puzzling pieces for cold seeps that are patchy, and they're not continuous. They're, as we said, oases in the seafloor. They're separated, sometimes very large distances, one from the other. And so one of the aspects to understand is how do animals move from one cold seep to another? Whether they move, how long does it take? How do they move from one location to another? So that's often one of the questions that we ask, because, for example, if a seep shuts down, which it may if the methane flow stops, then a new seep may be created elsewhere, and then we need to know how do those special animals get to that other seep? How do they arrive there, and how do they survive there? So all these questions are usually answered with experiments. Another so an interesting aspect of this sort of what we call those connections that happen between those patchy habitats is the way these connections will happen for animals that don't swim very far or walk very far, things that are attached to the seabed or they're attached to rocks, is through larvae. And so larvae, just very much like insects, are an early stage. They're produced by these animals, things like the mussels that we will see and the tube worms that we will see. They're produced and then they're released into the water, and they travel around in the water column with the currents. And so this is how an animal that cannot move will arrive, colonize a new location if those populations are patchy. Now, there are some animals that we only see in very specific types of substrates that they attach to. And so those particular substrates is bones. So we saw 3 days ago, or whenever those dive was, we saw a ray fall, and we saw animals that are on the were living on or sort of feeding on the ray fall. But there's also a specific worm that will colonize bones, and we don't see that worm anywhere else. We only see it on bones. The same happens with wood. If a piece of wood falls on the seabed and we collected one on our first or second dive, what happens is the wood sits there, and within some period of time, it could be months, it could be years, depending, this wood is colonized by these bivalves that we never see anywhere else. So we actually don't understand where does those bivalves come from, but they materialize. The same with, where do the worms come on those bones, but they show up. And this is not when the bone will fall, whether it be a food fall or whether there will be wood that arrives on the bottom of the ocean is not regular. It's random. It can happen at any time and at any place. Yet those larvae of those organisms appear to be in the water at all times, and they colonize. So what we want to do is we want to see the rate of this process. And so today in our experiment, we will put down pieces of wood and pieces of bone at two areas. One will be away from the cold seep in sedimented habitat that does not have seep effluence, and the other set will be put in the middle of the cold seep. And so there are some suggestions that some of these animals may be using other habitats like seeps or hydrothermal vents to sort of as stepping stones to go from the seep to a bone, etc. So there may be some of them may be harbored in these other chemosynthetic environments. And so this is why we're going to draw these comparisons. We're going to put bones in both areas. We're going to put wood in both areas, right in the middle of the seep and on outside the influence of the seep. And then we'll come back in hopefully a year or two, and we'll collect those bones and pieces of wood and try to see who has colonized them and if they're different animals that have colonized in the periphery as opposed to in the seep. So we're very close to our first location, which is outside the periphery of the seep. And when we get there, we'll start deploying the bones. So it's starting to get a little rockier. So maybe this is, I know we're not exactly at the site, but if we mark the location, we can always, come back to it. I mean, we just need to know where we are. We don't have to be exactly at that spot. And so here we're starting to see the sediment changing. Whoa, okay, so we need to, yeah. So we want to be able to drop before we start seeing, I think what we're seeing are mussel shells.
Good indication.
Yep. So we, if anything, we would like to sort of head back a little bit so that we have no evidence of any seep. This happened very suddenly. As you can see, it's a very sharp change. As we were saying, this is what makes it an oasis. And so we want to be away from the influence of the seep as much as possible. Obviously here, there's dead mussel shells, so there may have been a seep or they may reactivate a seep. So we want to be away from that. So if we could go back a little bit where we just had sediment without any evidence of seep organisms, to drop that first set of bones, then would be great.
Maybe stop the movement, please.
Could you move this up here so I can see that side?
Yes.
Please. Thanks. Perfect, thank you. Okay, so we are starting to see quite a bit of seep influence. Obviously, these are dead mussel shells. A little bit of discoloration of the sediment, so we'll move a little bit. We're moving a little bit further back to make sure we're completely away from the seep influence, and then we'll just drop them. So turn around them. So there is a strong current, and so the ROV needs to be, this looks good, needs to be positioned just right because we have to do a lot of, put heavy objects, deploy heavy objects on the deep sea bed, and there's going to be sedimentary suspended, etc. So we want, with the current, we just want to be positioned just right. So this looks great.
So you saw the heads of that 1309?
No, just we'll sit down here and then take out the experiment, one half of it. We need to mark the spot for sure, so we definitely need a waypoint once we sit down. Yeah, a lot of stuff on the porch. Perfect, this looks great. Okay, so this is, what color is the bar? What? Red. Okay. Yeah, sure, for sure. Yes. So the porch, this is not simple because we have 10 pieces of bone, we have 10 pieces of wood, and all of these have been weighed and measured because what will happen is as the animals eat them, of course, they will change in weight and size. And so when we collect them again, we want to know how much they have changed, how much has been eaten. And so they're all individually labeled and, the weight and volume measured. And so they have all been arranged on the porch in a particular way to make sure that we know which piece of wood goes where and which piece of bone goes where. So it's all sort of very organized so that we can identify every piece that we put on the seabed. And so what you're seeing now on the porch is the bio box. It has that one piece of wood attached to it. And so that will come off and we'll put it in place. And then you also see on the bottom there and a piece of a bone, which is cow bone, which is all the bone we could find. Now, typically, of course, we're not going to see cow bones at this depth, at this distance from Trinidad and Tobago. So typically the bones that are used, well, obviously that will be encountered with either whale bones or fish bones, fish falls. And so these are different. Obviously they're mammals, but they're also not exactly similar to whale bones, but this is the best we could do. And this is typically what has been used in other experiments. Other ways of doing experiments that have been done in the past have used, if there is a beached whale or a beach mammal or a seal that has come beach to shore and the skeleton can be quickly collected and deployed, then sometimes those are used if available. But again, this takes a major operation, major coordination. So it's rare. Most often, sampling is done on whale bones or skeletons that have been found by chance, and then a lot of sampling happens there. But again, that is more of exploration rather than actually running an experiment to try to understand how fast colonization happens. Are we ready? We're ready to go? Okay. So what we would like to do is we have 5 bones and 5 pieces of wood that are red? Red. They have red tape. They're marked with red tape. And so these are the ones that we want to place. So it doesn't matter what order they come out as. It doesn't matter. They've been here. They will be here for a long time. We don't want all the bones over here and all the wood over there. If possible, we want to intersperse them. So we have, a mix and match. And they best not to have them right against one another. But if they're separated by, you can put a few and then, within, I don't know, like 2-meter area. So you're gonna have to move. Yeah, we don't want them on top of one another. So, let's just start putting some out, about 1/2 meter separated and see how it goes. Okay, here it goes. Yep. So you can see the tags that are on each one of those bones because they're all identified. They have a code and a number that goes with it, so that then we come back and pick them up. We'll know which one weighed how much when we put it down. Ooh. I think so. Yeah. Well, I'm not. We'll take a picture of all of them all together.
So this is all one shot. Yes, it's just then there's survival. Okay. And then it's bones. And then yeah, and then this is
The numbers.
EFCB 8 and then
Yes, those are the numbers.
okay. So I take a snapshot
And
now.
this will be back.
Okay.
Yeah, I mean,
Survival,
that's
you
fine.
said? Colonization.
Colonization. Yeah, that's
Okay.
Yeah, those are good distances. Just spread them out like that.
Did anybody see what number that one was?
The one far?
The one he's holding right now on the left.
Yeah. Oh,
10. Got it.
really small. Yeah, just toss it. Goes. The good news is we don't have to be particularly gentle here, so. Yeah. There we go. I mean, what's the distance between them and yeah, that's perfect. Yeah, just in this. Yeah, but then keep in mind that then we'll put the wood in between. We don't want all bones, all wood. Yeah, yeah, perfect. Over there, perfect. Just
What number for this one?
that's 8, I think. Yeah,
Got
8.
it. Yeah.
Sorry.
Why am I saying 4? I see, perfect.
What do you want this one?
Just anyway.
As far as we
Sure,
can.
yeah, just toss it. I mean, it doesn't matter. That is
This one.
number.
Oh, 9.
9. Number 9. That's fine. What? So the other one is where? It's behind there, is it? We have 5. We'll take a picture when we're finished, when we go up. Okay, so now we need to get, oh, yeah, there it is. So now, that's the bones, and so we'll now spread, we'll put the wood out, and then we may need to sort of, I don't know, how to just sort of shove them a bit when we move back. I don't know whether it's easier for you to, like, these 2 bones are pretty close, so that would be.
You want this one close up?
I want that one closer to the sub. Yeah. Please. Yeah, sure. That's good. I don't want it stuck on it. Drag it over the whole knife. Okay, that looks good. So now we'll put the wood in the midst here. 5 pieces.
The bulge is gonna push them.
Well, can we move out a bit and then we can just a little bit and then. So we're not, yes, there is an explanation about what we're doing here. We're putting down 5 pieces of bone and 5 pieces of wood, and we're gonna see, we can return and collect them in a year or 2 years' time and try to see who colonized them. Because these are particular animals that colonize wood and particular animals, different animals that colonize bone. And so we're not going to bury them. We're trying to imitate a regular foodfall that you would see in the deep sea or woodfall. And so we're just going to leave them at the surface here of the sediment. And they're not going to drift because we're going to mark this side because we have to be able to find them again. And so you can see they're all labeled, they're all weighed down, they have weights, and so we hope to when we come back, that we will find them again. Otherwise, we will not know who colonized them. So no drifting, hopefully.
I've never seen it.
What about here? Sure. That's good. Yep. Perfect. Yes. So this is obviously wood. 9. It's number 9.
Got it.
Sorry. Yeah, I'll just point at those. Oh, it's fun. Isn't this fun? So you can see here below the wood, we have these little packets of weight that we have put so that the weight does
Yeah, just about
where the wood doesn't float up. Just perfect. That's fine. Don't worry about it too much. Okay, so maybe we can move on a bit. Pick up our plastic. Oh, sure. Johnny goes under here. He goes somewhere else. So we shouldn't go too far because we don't. But that's okay. Don't worry about being too precise. As long as we'll take a picture in the end, so we'll know where we put them. So it's not. Doesn't matter really. It's underneath us.
Oh, that's gonna be interesting. When's that?
5 bones, 2 pieces of wood, right? So we have, yeah. So 1, 2, 3, or whatever. Okay, so we have the last set of bones. We put them here. So you can see that the sediment where Sebastian was sitting before, you can see that it has dug up the sediment below the surface, and the surface is this brown, but below that is darker, which suggests that there is some chemical reaction oxidation processes. Maybe there's not enough oxygen out there below the surface of the sediment. But there's certainly a very thin layer of oxidized sediment at the surface, and that's mean it's well oxygenated, and it's the brown color. Yep. There we go. Just drop it. Don't overthink it. Just drop it. It's okay. Yeah. That's great. Yep. Fine. We have one more. So that was number 4. What number was that?
Looks like a 7 or a 9.
What number was that one? Couldn't see. Yeah, when we get up.
This one coming out is 10.
And that's all of them, right?
No, there's one.
Oh, there's one more. Ah, yeah.
That's a cycle.
Eight. And this is why we're saying it's tricky how the ROV will position itself, because you can see there's a lot of sediment, there's current, and it's hard to see what you're doing. So particularly today, we have quite a bit of current. So it makes it a little bit more challenging, but not for our pilots. You can put it off to the...
That sounds like
Yeah, sure, yeah. Perfect. Great. So now everything is in place. Will be good to take a...
Is there one more?
What's that?
need the number for this one.
Do we need the number?
There's a fifth bone that moves.
Seven.
Hold on.
Oh, it's there. Okay.
Seven.
Yeah,
Oh.
I got it.
We got it? Great. So where do you folks like site markers? Do you like in the middle of this, off to the side, like a marker?
Better.
Okay. So there is a site marker that we will need to deploy. So the site marker will be useful for the pilots to find the site. I mean, they will have the general, we'll get a waypoint and we'll have the general location, but the precise location will be much easier to find with a site marker. And you think that may be something fancy, but really it's a bucket lid with a weight and reflective tape. But because of the reflective tape, you can see this bucket lid from far as you're traveling along the seabed. Here we are. Ooh. Oh.
There's a flyer for you, right? So just drop it in the middle.
Yeah, but we don't want it to interfere with, so like not on something, maybe. If you don't mind, I don't know how far there you can reach or
Put it up for this.
maybe, yeah.
On board.
Either
Yeah,
way,
sorry.
there.
You need to, you need to line.
I don't want to disrupt it.
Just the camera straight.
I don't know how far that is. I don't know how far we can reach. Is that, can you reach there?
Can you reach just to the left there? Hold. Swap arms, guys.
Yeah.
I'd rather it's not right in the wood, so if it could be there. Is it strong? So just, yeah, it's a clutch. Yeah, perfect. Just perfect. Great. So now if you could, we would need to take, yeah, just,
The
yeah,
area.
lower enough. Yeah, so lean back and let's just, so we can see the arrangement, take some pictures. Just have to wait. Again, there's a bit of current, it's a bit of sediment. is not easy. Okay, so I'm saying, oh, there's the other bone. Okay. Yes,
Oh, over
please.
there. I didn't see that one get dropped. The one that's over here. Do you know what that one is?
What?
This one.
That's good. Okay, could we, if you don't mind, if we could, yeah, put perfect the lasers, just so that then when we estimate how far they are from one another, we know what that distance is. That would be great. Yeah. Perfect. Perfect.
It's okay.
Yeah. Great.
What's your next?
What's next? Okay. So I guess let me check. So we can sort of start. Could you zoom out a little bit so I could see the waypoints, please? When we're in that, we were running into, I think, yeah, we don't need to go directly to 1309. Let's just head, yeah, exactly. We don't need to fight that current. Let's pick up that line to the waypoints as quickly as feasible, given the current. Okay, so our experiment has now been deployed, half of it. We will repeat the same deployment set of 5 bones and 5 pieces of wood and a marker in the middle of the cold seep, the methane seep. And the idea there is to see if there is any differences, if we see different patterns in colonization, only because there is a suspicion or hypothesis that the animals that colonize bones and wood may actually be found in the seeps. And the reason for that would be that when the bone and the wood start breaking down, they also release gases in the sort of similar kind of chemosynthetic environment as we see in the cold seep. And so it could be, that there's some connection there. And so we like to test that. So now we're heading up towards from the periphery. So we were away from the influence of the seep. There was no evidence of alive animals, seep animals that we'd expect. And so we're heading towards a location that we know has been active before. And we'll see how that change happens from the background communities into those very different communities that we see at methane seeps. And again, you can see it different from the sedimented environments we saw in earlier dives. Here, as we approach the seep, we start seeing shells of dead mussels. And, there could be different reasons for this. One is most likely that those mussels maybe rolled down from above an active seep, or it could be that there was some fluid seepage here in the past. It stopped, and then those mussels died because they didn't have access to the symbionts that need that fluid to chemosynthesize. So a lot of shells. So that's me for today. So I will now pass it on to Jamie, she'll take you on to the next hour into the cold seeps. We do have a waypoint for where we were. We can add a waypoint when we know where we are. So can we add a waypoint there that actually says periphery experiment? Thank you.
Yeah, so just narrowly out for seeps. We're going directly so we get there a little before because we're still there and can't pick up.
All right, good morning. I'm Jamie, a grad student at the University of the West Indies, and I'm going to take you through the next hour. So we are currently making our way up Dome 1, I believe. And so you may have heard we are exploring known seeps today that we have seen twice before. So the first time that we saw these seeps were in the 80s by a French vessel. And then we came back in 2014 on the EV Nautilus, where 2 local scientists were on board to revisit these sites. And now we're coming back for the 3rd time. So we're in a general area called El Pilar, and we're currently on Dome 1. Dome 1 and Dome 2 we have seen before and twice, and in 2014 we discovered 2 new seepage areas called La Jablesse and Marmadillo, which I'm not sure if we're getting to today. We may, if time permits. And so we're currently making our way up Dome 1 back to the known seepage sites. And I mean, it'll be really interesting to see, because now that we have these sites documented over a scale of 40 years, we can see how the communities have changed. You see there's hard rocks. We can expect to see a lot of mussel aggregates, a lot of dense areas of dead mussel shells, which a lot of animals like to use to form their habitats. Oh, look, I'm seeing a skate, I believe. Yeah. Yeah. Can we zoom in on the skate? Okay, so these are clam shells that we're seeing here, not mussels. And we actually have a local scientist not on board who studies skates in Trinidad and Tobago, so she's always excited when we find footage of one.
berms. See the two berms?
Yeah.
So they are. Two berms. Yeah, I mean, we have one on a general. Oh, we're seeing two berms. We're getting close. Oh, we are they? So many two berms. Bubbles. We want to go. To the left. Yeah, to the left. I believe.
Sure that's not.
No, it could be
Ross.
sediment. It could be through. We're gonna see.
That's coming out.
Yeah. It looks shimmering.
I was gonna stop at that more here.
So we're just waiting for sediment to clear a bit before we get a better view.
Just looking around here.
Yeah. We want to, yeah.
So we can stop in here.
Yeah, just for a bit.
Bridge, you can come to us, stop there.
Coming to the sun.
It's a lot to see. There you go.
Oh yeah. From the where we had the bubbles. Yeah, sure.
Yeah. I mean, I guess we can think our way to where we want the bubbles to be. We can see coral along the rocks. Yeah, we're going up. So we would. She wants to go not to 3, but across to 2, which go a little bit further.
Go to this.
We'll go to 2 first, and I think we can cross. And then we can drop off the rest of the ones in the wood. And then go back up and explore 3. We can't really pick up anything. It's all going to the wood.
Good, we got something. So we got straight to
So we're on the straight to 2, but
So you don't want to look at these areas where you see bubbles or stuff.
yeah, we're gonna do that after. After. I don't want like that way. And then back. Sorry. Yeah. Look at all these tube worms. So what's happening here is because we are on the Barbados accretionary prism, where the Atlantic plate subducts under the Caribbean plate sort of scrapes the Atlantic plate, and then that scrapes off the top layer of sediments. And in that sediment, there is fluid, rich sediment that gets trapped and creates these reservoirs under the ground. And here, what we're looking at is where there are little cracks and fissures, hydrocarbon-rich fluid is able to escape through the seafloor. Ooh, wait, yeah, we want to look at that. The chimera.
Take it with the boot.
Oh, let's get this boot. Oh, he's running. Oh, chimera. Yeah, a ghost shark. Oh, he's running. Oh, howdy. He's hiding. Yeah, so the hydrocarbon-rich fluid, when it escapes from the seafloor, small little bacteria that lives inside of mussels are able to then utilize the fluid to create energy and start a food web. And then the mussels that have the bacteria create this hard substrate that other animals then rely on. Look at this. And so this whole ecosystem is mainly fueled off of chemosynthesis and not photosynthesis. And a lot of species found at seeps are actually endemic, which means they're found only at seeps and nowhere else in the world. And tube worms are indicative that we're on a seep. And so because we have video footage from 2014, we're actually going to be able to go back and compare the two sites and see how they've changed over the last 12 years. And then these are called coal seeps because the fluid that comes out is cold versus hydrothermal vents where the fluid is heated like to insanely high temperatures. Mm-hmm. Got it.
Got it.
Can I actually zoom in on the coral on the left? Those are big spot lobsters.
A little bit. Thank you.
And so those long tubey looking things are what we call tube worms. And there are a lot of dead clam shells all on the floor. Oh, he's looking right at us. Look at his face. Oh. We can see that. So when they came in 2014, they actually saw an octopus that they believe is new to science and never been seen before. So we are also looking out for this octopus. It's of a granulodon species. Oh no, that's a yeah. What you? No, down, down, down. That thing. How did that? Yeah. No. That's an octopus. That might be the same one. Hold on. It is.
Oh yeah, down there.
Yeah, see, I got good eyes.
Yeah.
It might actually be the one that we're looking for. I'm not sure. Alright. Let's go down and look at him. If it is. Yeah, we want a good look. Oh. Look at him. Oh, look at him move away. I think that's good. Hi, guy. Oh, no. He's hiding. See him come around. Oh.
You could come to us though.
Come on. Come on to a stop.
He's smart. Oh, there he is. Yeah, I don't think it's the octopus. Oh, he's. Yeah, the one we're looking for is purple. I think we can move on. I think we gotta go look at him. This one's a normal one. The other one's purple. thought it was a rock. I'll drop it off. Damn. And if we ever see if we see like a huge patch of tube worms, I'll let you know what to do, but we'll drop a waypoint because we're not sure what's up ahead. I think things could have changed in 12 years. Oh man. Oh, was it? I got some hints. Is it shimmering? Can't tell if it's shimmering or if it's setting.
It's backwards a little bit.
Yeah? Are we able to go backwards a little bit? Okay. Just giving you practice. Rob, you can point it out to me when you think you see it. Need to go down closer.
Like it's right at the bottom.
Oh, like right under hay.
You see this stuff that's waving?
Yeah, it's waving. The only thing I'm shocked about is that they're not like a bunch of tube
Or is
worms.
it like just the current going through there? Oh, maybe.
Do you think it's just the current?
No, that'll be the
False alarm. Okay, we can move.
move.
Yeah, we can move on. Yeah, because I was thinking if it was shimmering, there would likely be like a lot of
Things there.
things around it.
Yeah.
Tube worms, mussels.
Very could resume more.
Resume in a minute.
Oh, that was a big crab. Can we... Yeah, I just want to see what type of crab it is.
Yeah.
I don't know. Look in between two rocks. Okay. Oh. We're making our way. When we get there, we should hopefully see like mussels as far as the eye can see. Ooh. Yeah. It's shrimp. Shrimp.
Someone trying to stop.
Oh. Do you want to zoom in? All right. I think that's looks like a mass of dead tuberums. We could zoom in on what that is. I don't know what it is. No, actually, it's not a rock. We can zoom in on it though.
there's mussels.
Mussels. Zoom in and snapshots. These are our first live mussels. We're getting close up. I mean, there's probably bubbles somewhere there. These are gigantitas
Yes.
childressi.
Yes.
I don't know how
change
to-
the name.
Yeah, from Bathy modulus.
Oh, I see. Oh.
So if these are clatterizans, they're only known from Barbados' seeps, which are like 3,000 meters deeper. So
Look at the coral.
this will be the first ever record. Yeah. So,
Or
or just grab a clump.
if we stick it in, we have a location.
Yeah. So put a waypoint for sure also, but I think we definitely want to sample this just because
Okay.
it's the first. Oh, and live Bathy modulus.
Yeah.
Yeah.
Gigantitas,
Gigantitas.
gigantitas.
Sorry.
Yeah, we want to take a sample.
Oh, that's nice.
Yeah.
So probably in the quiver.
Oh, and they have the other sponge that's really. we wanna- but it's
In a
like
quip.
little.
Where? Yeah, we can put it in the quiver.
Yeah.
Okay. How much do we want? Like a clump. Clump.
Sure. Yeah, just show the clump. I mean, it's fluffy, so.
Oh, just this is flying.
Show your hands counting. We wanna. Okay, so we wanna drop a waypoint here because this might be the first time this sponge is seeing our waters. Very exciting, well. And then we wanna get a clump of it like this big in one of the quivers. Yeah, lasers, zoom up images. Lasers are on. And then we want to zoom up as much as we can. It's the whole show, the mussels, the tube worms, the sponge. Everything's on this.
Wow. Okay.
Mhm.
You can.
Yeah.
I think it's a stone not to pour something.
There what?
So it's more.
In the back. Oh.
No, it's a sponge. It's a sponge.
Ah, it's a sponge, yeah.
Get excited.
Sheesh. Oh, and a little, oh my gosh. Cal, what's the name of this fish again? Oh, I have it. My favorite fish. Oh, a Pachycara carabium. He has a cute face, but his body makes me a little uncomfortable. It's a little long. It's an eel pout. Cal, it really is everything. Everything we want. In one shot.
So right, it is, right? So okay, so I think what we should do here is we should get a sample of the coniferous sponge because it's never been sampled here. It's first record of it. The other stuff we should see at the other seep. Like this is a teensy little patch, right? So yeah, all the rest of it should be elsewhere.
Okay.
But just because this is like so weird first one, can a sponge range be like 5,000, like 3,000 meters? Can a sponge range be like depth range be like 3,000 meters? Yeah. Okay. Because they're known from the Barbados seeps where it's like nearly 5,000 meters.
Yeah.
So that is the one that we tried. Do you remember with Anna Reed? But when we get to the active
Oh, look at
seeps,
that.
it's there. So we can leave the sky.
Uncomfortable.
But they're everywhere.
The fish too. No, because I mean, where is it?
So I don't know if the name
The
has
two
changed
bums
now,
really
but
make
it
me a
was
little
described
uncomfortable.
our little seeps, and it's planarized and with plan of yeah. If that is what it is, if that's what's known from not that far away. And in Barbados, it's literally like bushes. No, they're totally are. Yeah.
Perfect.
So,
Just this one.
fourth of them,
Yeah.
sponge.
Yeah. How much? You can. Wait, no. They're debating. Yeah. Which Bible is open? Yeah.
Free. All of this one.
Yeah.
I don't even have a. Did you even give me a cup?
So we've emptied this one. So that's free. Leave that free.
And how much would you like?
I mean, we also want to like this is kind of
Yeah, I mean, like the right hand side, like the entire thing, like that amount.
Hey, use it. I can't see.
It's covered now.
Okay. No, is it on that?
It's yeah. I don't want this.
No, I know. It's because the box is open.
But
because
that
of the
amount,
water.
yeah, like that amount, yeah. I am absolutely only talking to Jamie, and Jamie's relaying it to the pilot. Yeah.
Mm. Yeah, yeah. Can I see the sheet again? Sorry, there's no sample sheet up here. I have no idea what box is empty or what's full.
This is free.
Yeah. Okay. So then any of these is fine. So all of these are free.
Just use a pen.
All of these are free, so we're just gonna put it in
And there's no water attached to it.
there. No?
No, this is all free.
Yeah. So any of these is fine. Now we're just going to take a small sample of the sponge because we believe this may be the first time that we're seeing it here in Trinidad and at this depth range. I believe it's a carnivorous sponge. And yes, a lot of these animals are typically found at chemosynthetic seeps, the tumors and the mussels, which is not shocking because we are going to a known seep site to see how it's changed over the last 12 years. Okay. All right, handing over to Kyle.
Okay, hello everyone. Thank you, Jamie. My name is Kyle Foster, and I am a postgraduate student at the University of the West Indies St. Augustine campus, studying deep sea biology. And excitingly, we are exploring some known methane seep sites today. And while traversing this ancient seep, we have found quite a lot of diversity within this one area. So we're seeing some sponges, some clams, some tube worms, along with some amphipods and other crustaceans. So we're just going to go about getting some samples, especially of the sponge, which we suspect might be Cladorhiza methanophila, which is a species only discovered and described in waters of Barbados, which of course isn't very far from where we are, but it hasn't been described anywhere else. So we're gonna get some samples. And can we zoom in on the sponge before we take samples, please? Thank you. So we're just getting some close-up shots. You can see lots of what looks like squat lobsters in and among the sponge and the mussels. You getting pictures? Yeah, of course. She's getting some frame grabs. There's quite a lot of excitement. Yeah. That's good, tree. Yeah. Okay. And now, yeah. Well, that's good. Thank you. We'll get some samples now.
Okay, so a piece of this into full cover number 1. And no, bio-box.
Bio-box.
Okay.
Should we switch bio-boxes? That was 3 again. 3? Bio-box 3?
3A.
3A. Okay. Spaghetti. Okay, then we'll just get a frame grab of it in the arm. That? Oh, can we zoom in a bit more, please? Thanks. All good? Okay. Good, thank you.
Okay, what's going on? is 3.
Yeah,
yes.
baby.
Yeah, that's fine. Got it now. Roughly know.
What you want to have further?
Okay. It's quite light and springy, it seems. Oh, it's blowing. Oh. It's very light. Almost like a feather. Oh, it's going. Oh. Okay, yeah, so clearly this sponge from observation is very springy, clingy, and light. Okay. Oh, we'll just zoom in and get some photos of them. Sorry.
Should have remembered this one.
Sorry. Okay. And then we'll just try to get some more ones here.
You want some more?
Yeah, well, once you all
Maybe we'll pick some of it.
are able to get it in the first place, because it's very light and it just blows away. Yeah, because I kind of the other part blew away. Wait, yeah, we're gonna get some.
This one. We're going to get a different chunk and we're going to put it in the big one,
Okay.
if that's okay.
Yeah, that's fine. Whatever is easiest for you guys, really. Oh. I'm gonna try suctioning instead.
Might as well take a picture.
Yeah. picture 6. But yeah, we're gonna have to try to get a lot more than, like, you know, the size that you had initially. That's like a good.
Yeah, so your place are trying a bigger piece. We're just gonna get like more after this.
Yep, get one out of the box.
Yeah.
I think that's fine. You want to put it here?
Okay. We're just, this one in here is fine, but if we could like just section some more instead of being raw, then.
It says description. Go through this.
Do you guys are able to choose that goes into that bottle too?
Yeah, yeah. I'll get into number 2, so we'll keep number 1 as a flushing bottle.
Mhm.
So we'll put it into.
Alright. very much.
What is that?
Nice, thank you.
I know. Everyone that would need that. You're on your own science. Yeah, fly, do whatever you want.
Thank you very much. Okay, so we'll just continue to explore and take closer pictures of things as we make our way to waypoint 2. But yeah, that was quite difficult to sample there just now, but thankfully we got a good bit. And yeah, right here in this screen, you can see quite a large diversity of animals. So we had the Lamelia brachia, sorry for the pronunciation, tube worms, and then some mussels and some sponge that we hope to be able to identify further once we have it with us. So yeah, so currently we're exploring an ancient seep, and then we're gonna make our way towards waypoint 2, which should be an active seep. And then you also might be noticing a lot of discolored patches on the sediment. So those are bacterial mats. These bacteria are what would be transforming the methane into energy, into food for much of the organisms found within this type of environment. Yeah. Oh, big fish there on the right. A Synaphrobranchid, maybe? Yeah. Okay, yeah, but nothing we haven't seen before. So yeah, so we're exploring the Barbados accretionary prism, which is a formation that spreads from Trinidad and Tobago quite far up the island chain in the Caribbean. And of course, Barbados being the aerially exposed part. And all throughout this accretionary prism, you would find bubbling of methane and mud volcanoes, and then associated communities that utilize the chemicals that are being emitted for energy.
Seep. We went to 3.2, right? And one thing, current is the seep. It's fine. I will tell you one way so to get that one. Love this red soap.
It's been easier to fly the closer we are.
I know. It's more risky. That's why it's called red soap.
So we're making our way up a slope. Lots of bacterial mats on the sediment. And the current is quite strong as well, so our skilled pilots are fighting the currents as we make our way up. Lots of clamshells around as well.
Terrible.
Octopus? Oh yeah, there is an octopus. We just passed down below. Good spot, Cherise. Thank you. Is this one more purple than the other one?
Is that the octopus y'all were looking for?
We saw something similar this morning. It was more red. This one is more purple. So possibly. Let me ask a question. Yeah.
You gonna land for this? Some pictures?
Oh yes, this is it. Okay. The Granuladoni. Nice. Cherise? Good job. get some close-ups of close-up shots. Maybe with some lasers on as well, so we can get a sense of scale, if possible.
My band.
Yeah. There you go.
Yep, there he goes.
I don't think he likes us.
Like, okay, bye.
Alright, well, we're gonna have to reorient, I guess. Went into hiding.
We'll go. Did we get all of our sample things off of us? So this, We still have some. starboard look still has some. This is empty.
This is pretty.
Because we didn't do site 2 yet. And site 1.
I think we shoot this one and then we sample it. No, no, it's just below us. Sure, yeah, just because I think. Well, let's just do that and get it out of the way, and then, but we can shoot it every single one. I do think we'll see.
We just take a look at where it is and what it looks like.
Yeah, and we can drop away from here. But we're not gonna mess it in the back.
So the sponge just went into a little bit of a hiding place behind a rock, and we're gonna try to see if we can get a better view. I mean the octopus, sorry. The octopus. Oh, here we go.
That's fine. Description, we'll just, it's gonna keep that description.
So of course, as we said, there's a strong current, so
Yeah.
whoa.
Um,
But yes, we have some skilled pilots on board, so we're in good hands.
no, sure, yeah. Yeah. But then do you know where it is?
Ah, yes, perfect.
Totally, but we're not gonna be able to work on it. Every single one. We can't shoot every single one. So do you want to film this one? Or do you want to try and wait?
Ah, it's a nice shot.
I think the call will be on the current and how stable it is.
I feel like we should do this one.
What are your thoughts on the current?
I agree.
Racing.
Pretty crazy.
With the ally. Yeah.
Oh, nice.
You on the bottom there?
Just hiding.
So yay or nay? Don't do this on my behalf. So we're doing it?
Oh, wait.
It's pretty crazy in terms of current. Stability is going to be tricky, is that, yeah?
Yeah, I'm working on it though. Let me see if I can give a little more gallons and that doesn't. out like 85 since then.
Wow. So gorgeous octopus, which we suspect might be new to science.
It adores.
I know.
Pretty stable, there's that.
This octopus genus
Like
is nicknamed the
mentally
warty
or
octopus for obvious reasons.
The one
This
for
octopus
attack.
genus for now I don't is nicknamed the warty for obvious reasons.
Okay, so I've been informed that this species of genus, sorry, of octopus is nicknamed the warty octopus, and you can see clearly why.
So can you pay for me to take over camera?
Yeah.
I'm going to do a couple of shots without the light, and then we'll get the light just in case he goes.
Okay.
Thanks so much, everyone, for your patience. We have been engaged in a very exciting project, but now we're back to science. My name is Diva Amon. I'm the chief scientist of this expedition and deep sea biologist from Trinidad and Tobago. And this octopus that we've been spending a lot of time with, this is really exciting. So we observed it in 2014 during the Nautilus expedition here. And we had just 3 days during that expedition in Trinidad and Tobago's waters. And this was sort of one of the star observations. One of the foremost octopus experts in the world, Dr. Janet Voigt from the Field Museum in Chicago, she reviewed this video from 2014 and she is pretty convinced that this is a new species, a species that no one has ever described before for science. And so because of that, we came into this expedition very excited at the possibility of seeing this animal again. We only saw one on the first expedition. And again, it was just because we were only there for 3 days. But it's very exciting to return to these seeps 12 years later and to find another one of them. And so because it is potentially new to science, we are going to try to collect it. The reason we have to collect this animal is because it is impossible to do a scientific description, i.e. give it a name and, introduce it to the world properly if you do not have a specimen in hand. You need that specimen to be able to, look at all of the morphological characteristics, so the different features of its body, and compare them with other octopus that have been observed around the world. But then also we use the specimen to look at its DNA. We do molecular analyses, and that can tell us a lot about the animals, both inside and out, essentially. And so by combining those two analyses, we can then with absolute certainty say if something is new and then write the description and give it a name. So this is something that we are really excited about, and this octopus will potentially go a long way to helping us to understand more about this species. Of course, octopus are always really challenging to collect. They're incredibly intelligent, incredibly dexterous. And however, there's still enormous scientific merit. We do only plan to take one, at most two, a female and a male, if we were to see a male. We do think this one is a female. Females tend to have less. Oh, now that she's swimming, actually, I think I might see the appendage. So how you distinguish between male and female octopuses is that one of the arms on male octopus is a modified arm. It actually has a modified end and almost like groove that runs up the entire length of the arm that it uses to deposit sperm into the female. So essentially it will use its arm and the sperm will shoot down the arm and it will be able to put that sperm packet into the female. So this one actually, now that I've just been calling it a girl for the last however long, but I do think now it's a boy. And if it is a boy, that's actually potentially a bit better for science. Because they tend to have more distinguishing features. And it's absolutely not easy to collect these. It just shows how skilled these ROV pilots are on board. It takes immense skill to be able to do that. And this octopus will serve enormously for science and for outreach as well. And we hope that this is just one of many new species that we are collecting on this expedition. As I know many of you are very regular listeners, but Trinidad and Tobago's deep sea and the Caribbean deep sea in general, and actually the deep sea in the Caribbean is about 86% of its area. So thank you so much, guys. That was great. Is really, really poorly explored and therefore poorly understood. We know very little about the ecosystems, the species that live down here. And so these collections, I mean, this octopus specimen that we have on board now will be the only existing specimen of this species, potentially. So very important, and will be used as a basis for lots of new science in the future. Thank you, guys.
Thank you.
Okay, so we would like to continue to waypoint 2, please.
Waypoint 2, copy that.
Yeah. So we are on a big structure called Dome 1. It was visited by the French in the 80s and then again by the Nautilus in 2014, but for a very, very short period of time, just a few days. And this dome is home to a number of methane seeps, areas where bubbles of methane gas as well as liquefied methane are seeping from the sea floor, and that is then powering ecosystems down here. Many of the animals that live at these seeps are chemosynthetic. That means that they have symbiotic relationships with bacteria within their bodies that essentially use the methane as a form of energy. And because that energy is so plentiful in sites like these, it means that you get a lot of life all in one area. These methane seeps tend to be oases of life, and that has led to some of them around the world being designated as vulnerable marine ecosystems by the Food and Agriculture Organization, FAO, by the Convention on Biological Diversity. Some are known as ecologically and biologically significant areas. These are special places on the seafloor with very special animals. So someone is asking how long a specimen like this would remain in a collection to be useful for science. And I mean, it's in perpetuity, ideally. We have in museums around the world specimens from the 1700s, 1800s. And of course, our ability to understand how to preserve things is always getting better. And so the lifespan of many of these specimens that are collected is very, very long. And as science and technology grows, we find new ways to be able to analyze these specimens in the future so that we can answer new questions about it.
Go ahead. Can resume the vessel move for the position. Thank you.
Yeah, that one was. So we're on the way to our second waypoint on this dive, and we have been mapping all night. And we've also have bathymetry and other types of data like bubble streams from in the mapping data from 2014. And so we mapped overnight and compared our mapping data to the 2014 mapping data. And we did, in fact, again, pick up bubble streams here, which indicates that the seep is very much still alive, or the seeps here are very much still alive. And so we are on our way to waypoint 2, which is where there are a number of seeps that have not yet. Hold on. Before I say the wrong thing, let me check the dive plan. So we're on our way to dive 2, which to waypoint 2, sorry, and that is along a trajectory which has a number of previously active seeps, a number of places we have seen bubbles actually emerging from the seafloor, which indicates that methane is seeping out, and therefore we expect there to be a very high density animal communities in these places. There are also on this dome-like structure a few areas where there are bubble streams that have never been visually confirmed. So this dive is a little bit of both exploration as well as revisiting very special places. And so one of the RV pilots was asking about the terrain that we're seeing. So these rocks are carbonates. These form during the sort of seepage process that's happening, and you are going to have Rachel Lauer join you in a little bit, who can speak more, much more knowledgeably than I can about the geology of the area. But you'll also notice in between the carbonates, there are these white shells. These are bivalve shells, so they're a type of clam. And these clams are known to be chemosynthetic. They have special bacteria within their tissues that allows them to use these chemicals seeping from the seafloor as a form of energy. And these clams tend to be buried, so they can be quite hard to find. But we are seeing, when they're alive, but we are seeing many that are dead on the ground. But again, they're a really good indication that chemosynthesis is still alive and well here. And we can see that some of the shells on the seafloor are sort of coated in like brown stuff, there's some kind of brown coating on them, whereas we can see others that are white. And so we can expect that maybe there's an age difference in some of these shells. Rachel was saying earlier that her understanding is that this entire area is an ancient cold seep. These have likely been here for millions of years. Which is really cool, and I'm sure she'll tell you a little bit about that later. When we were here in 2014, we did not do very much sampling, and so we're planning to spend today's dive at this particular dome and then tomorrow's dive at a neighboring dome where the animals are broadly similar. And so collecting samples, specimens, will be a really important part of the day. Of course, there are many others, but it will be a really important. Ooh, sea toad.
Sea toad?
Wow.
Isn't it sea toad? Chonax? No?
That is beefy.
Goosefish?
Goose, maybe.
Goosefish?
Yeah.
That guy is interesting.
Yeah, and if you go closer, they have a little lure, and they walk on their little feet.
Wow.
So cute.
Wow. Yeah, it's the old lure on his nose.
Do we have any more zoom?
Oh, yeah. You got a lot. You want more?
Yeah,
Can we put lasers on?
just a second lasers.
You want me to sit down so it's like super stable kind of thing?
I mean,
These little lasers. He's pretty
Oh.
decent.
It's cute.
oh yeah, he's chonky. This is a chonky boy.
Like a watermelon. It's bigger.
But yeah, you can see it's got a little lure on its nose.
Why does he remind me of Chimp?
Keep the laser on.
So you can see the lasers have just come on. Those are 10 centimeters apart. So you can see that this is not a small fish. You can also see that they've got modified fins so that they can actually walk along the seafloor on, as well as all of these little baubles that I assume they use for sensory purposes dotted all over their bodies.
What a cool dude.
Very cool. Are you guys good for video?
Yeah,
Alex, or you want more?
I mean, I'll probably take more, but
Lol.
we've got good footage.
I can park and like super zoom on him if you
I
want.
mean,
You're the boss.
I think we're good. Let's keep going. Just
Fine,
let's get to
yeah.
those seeps,
Yeah.
please.
It's good.
But it is cool.
It's super cool.
That's fun, guy.
Look at his face. Look at those lips.
Oh, kicking the thrust.
Quite a large. Wow.
Okay, let's go.
All right. See you later.
Great.
See you later, Jimmy.
Oh, yeah. Okay, that's it for me. I'm going to hand you over to Robert Cassidy and enjoy the rest of the dive. Thank you, everybody. That was an epic collection.
Thank you.
And the other things we were doing.
Hi everyone, Robert Cassidy here, PhD student at the National Museum of Natural Sciences in Madrid. And I'll be with you for the next hour while we continue exploring this methane seep.
Is that old worm castings? Oh, it is
Yeah,
feather worms.
those, yeah. What are those? Oh yeah, yep. Wow, so we got some feather worms here. All these tubes that you see those are created by these polychaete worms. Very weird looking animals, and a lot of these types of worms are actually associated specifically with methane seeps and like this one, as well as hydrothermal vents, what we call chemosynthetic ecosystems. Yeah. Could you maybe zoom on that one too? Just to get a look.
Okay, sure.
Wow.
You can, when I get it centered, you can go ahead and zoom in. Wait till it's centered. Okay, go for it. Perfect.
Yeah, so these things that look kind of like anemones, they're actually they're like the mouth parts of these polychaete worms that stick out of their tubes, and they kind of collect things that are drifting by. So when you see them contracting, coming back out again, that's actually them, you know, trying to eat something they might have caught floating by. Yeah. Wow.
There's a different kind there on the bottom left. See that?
Yeah.
Same kind.
That is different. I'm not sure. They've got those red kind of feathery bits on the bottom left, and there's that giant tube as well. That might be a different kind. Very
Carry on.
cool. Yep. Thank you. Yeah, so for those of you who have been watching along the past few days, you might notice that this is a very different looking type of seascape we have down here.
Can you move the boat a little bit to starboard too? Just tell them to come about.
That's because, you know,
Starboard while they're doing that.
a lot of these animals have to adapt to these specific conditions, like anoxia, which is the lack of oxygen because of the kind of unique chemical aspects of being by a methane seep.
Movement, is it possible to go around 10 meters to starboard?
That means there is starboard, yeah?
And a lot of these animals not only tolerate habitats like this, but they actually can only live in them, right? Because many of them have very unique symbioses with different bacteria that actually will process these normally toxic chemicals coming off the seep and turn them into food for the host animal.
Okay, we'll just keep going for now then. Keep going as it is.
Cool. Wow, and you can see that some of these tube worms actually, they create these big structures that you can see on the bottom right here. You know, these actually are important for other types of animals that live down here as well.
Sure.
Yes, please.
So I've seen a lot of fossils of worm castings too. Is that this kind of
Yeah, you know,
similar animals that do that?
I think that would probably be similar. You think those are fossils? I wasn't sure if they were fossils or just like, you know, more recent but like left behind things. Could be fossils though, yeah. And seeing what looks like clam shells and things like that on the bottom as well. got a coral here. 2 corals.
Are they following the track?
I
Uh,
mean, we're following whatever.
The line that
Yeah.
they get it?
Yeah.
Yeah, great.
So as Diva was saying, this is actually one of the few sites we're going to on this trip that has been explored before. I think it was back in 2014 was the last time people were around here. But even so, these types of habitats are so biodiverse that you can't possibly see everything in just one dive with an RV. So it's very much worth it to come back and see what else we find. Great example being the octopus that we just were luckily able to get a sample of, which we believe is probably a new species.
These are all these worm castings.
Oh, you think so? Got it's either a crab or maybe a squat lobster down here as well. Got So it might be surprising to think that we were able to find a very large but yet undescribed octopus species down here. We have to consider that chemosynthetic habitats were not known to exist until the 1970s, right? And still not that many have been explored relative to what we think is out there. So it's still a very, very young kind of area for science. It's important, right? Because these areas with methane seeps, the type of geology that produces that is also very much of interest to oil and gas companies. So it's important to, explore the biodiversity of these places in order to effectively manage the way they're used for industry. Our shrimp hanging out on the seafloor. Do you have the sample number for the octopus? What's up? You say old coral? Yeah. Yeah. So our pilot Zach is saying that you might be looking at an old coral kind of on the bottom there by the rocks. Possibly fossilized, maybe? Wow.
Really interesting rocks, huh?
Yeah, very cool. So as we're moving along right now, we are kind of slowly working our way up the mound that forms a big part of this methane seep, gradually getting closer to where the actual seep might be happening, according to our maps. So I would expect that the fauna will probably kind of shift a bit in composition as we get closer to that site.
Giuliani, where are those waypoints on here you're talking about? So they're, okay, so they're markers. Do you guys want to stay on this line super close, or does it kind of meander as we go to two? Because we're going to pass right by one of those bubble sites they saw.
I think we want to stay on the line as close as we can.
Okay.
Yeah.
Sweep over more gun? Ah, yeah, okay. Spoon. Let me see the spoon.
The ice cream scoop.
So we've got something shiny down here, trying to see what it is.
Is it just the inside of a shell, or?
It kind of looks like the inside of a mussel shell.
Yeah. Yeah, it could be, yeah, the inside. Yeah.
It's got that kind of mussel shape to it.
Yeah. All right.
Yeah, so I think it looks like the inside of an old mussel shell. Mussels are one of the main kind of big habitat forming chemosynthetic animals that you find on seeps like this. So we will surely be seeing more of those, hopefully alive, as we move along.
I can never remember.
Got, looks like a big fish on the right, under a rock, kind of.
Yeah.
It's not too
Oh,
bad.
what?
It's fiber optic.
I mean, in normal circumstances, we wouldn't want to do that. But with the current, it's kind of, we have to be as close as we can.
We do too.
Yeah, the less, the main thing would be if I start getting like where the entire ROV is in there, let me know. But remember, the size of the ROV changes depending how much you zoom in and out too. Right? He's hiding.
He's kind of shy, this guy.
The Octa, remember the Octa was too.
Yep.
Almost like they've never seen an ROV.
Okay, we've got some squat lobsters on that rock as well.
Weird rocks.
Yeah, very interesting rock formations.
It's like entirely made of those worm casts. It looks like almost.
Yeah, it's crazy.
You see all the little
Yeah,
bits and bops of it.
yeah, you really can. I wonder where that kind of webbing is.
Spiders.
Yeah,
Sea
spiders. Got some, ooh, maybe some small gorgonians on the left here. Yeah. Wow.
There's a lot of stuff on it. Right next to me. Big thing.
Yeah, so this seafloor landscape is very different from what we've seen the first week of this trip, which is mostly like sedimented, relatively gentle slope. Here we're on a really rocky surface and a pretty steep slope as well in certain places. So as well as exploring the biodiversity of this area, we've also been doing some mapping during the night when we're not down with the RV. That's really important because, you know, even though people have been to this site before, the maps are still pretty limited. It takes a long time to get good maps of an area like this, and it's really invaluable for deciding exactly what areas you explore, right? We typically try to base exploration areas off of places that have more slope or rockier surface, because when you have a slope, you get upwelling of nutrients, which attracts more animals, and when you have rocks and hard substrate, you get more of those benthic invertebrates that kind of form the gardens and the, you know, the structure for a lot of different animals. for those of you who have not tuned in until recently, in just a few hours on this dive, we've seen carnivorous sponges, we think. We found clams and mussels that are probably chemosynthetic, and a possible new species of octopus, just in a short dive. So, just illustrates, you know, how biodiverse places like this can be. Some methane seeps have actually been found to have the same biomass as coral reefs in shallow waters of the same size.
Wow. You can see it. Look at all this evidence
Yeah.
of life. You know, the shells and the
Yeah,
worm castings and stuff.
yeah, it's very cool. It's especially cool because people used to think that there were no ecosystems that depended on sources of energy that were not the sun, right? But starting in the 1970s, and with the discovery of hydrothermal vents and the animals living on them, maybe the vents were discovered before, but I think they discovered the animals living on them in the 70s, which just opened up a whole new, you know, area of biology that studies animals that are dependent on nutrients coming from, you know, geological things like methane seeps and hydrothermal vents.
I mean, it's still pretty much straight down.
You've got a big field of, it looks like mussel shells or maybe clam shells.
This is that shiny thing you saw earlier, right?
Yeah,
Same thing. I think.
yeah, same thing as that shiny shell from before, but just a whole huge field of them.
Exactly.
Wow.
I don't know how close you want that to get to the hole.
You guys might want to note this in some kind of observation, I'd say.
Have somebody go look
Wait,
at it.
what?
I suspect that camera's.
I think the camera just gave it a little. Yeah.
You want a waypoint? Yeah.
I'll go. We'll go.
Yeah.
I think the camera angles.
Okay.
Tricky.
Yeah. So if we could try to zoom in on some of these shell fields and see if we find any living ones, that'd be great. As well as we drop a waypoint.
You want a marker here?
Yeah, that'd be great. Maybe once we're like, yeah.
You know,
I think
make
this
a
one would be
marker,
good.
right, Rob? Go ahead and make a marker whenever you're happy.
Yeah. It looks like some of these might be alive, like these darker ones.
Yeah, that's why.
Hard to tell.
Yeah. Definitely that.
You want me to sit down and stop?
She collects them.
Big pile.
Oh no, we're not collecting.
No. Okay.
Just keep going, but
Keep
if
going?
you get some pictures and mark it.
Yeah. So make sure you get some frame grabs if you can.
Okay?
Especially when we see like the dark ones.
No, I do. Not, it's inboard, but not touching?
Don't touch it. Yeah. So this is kind of what I was expecting. I think these mussels can live pretty close to where the seep is actually happening. So it's not surprising that we're getting these whole fields, because now we're actually, I think we're basically right on where the seep is. And these mussels are one of the best known examples of life on cold seeps.
I can't wait to get these ropes off the front.
Yeah. These are from the bone drops and stuff. Yeah. I did, yeah. like some kind of like microbial mat.
Yeah.
It's kind of a
No,
weird
we can
color.
zoom in to see, but
Yeah.
it's a little dark, but we know that something's happening.
Yeah.
Sorry to have disappointed you.
So one of the ways that we find these areas is actually by, well, I can't tell you exactly how it works, but one of our residents, our geophysicists, Rachel and Rob, will do these mappings that actually can visualize bubbles coming up from the seafloor when they pass by. I think it's on the multi-beam. So we actually get maps of exactly where these columns of bubbles are coming out, which, you know, when you get a column of bubbles in the deep sea, it tells you that something is going on. It doesn't just happen, unless there's some kind of a unique geological step happening, like a seep. Whoa. Looks like, yep, chimera. Was this a ghost shark like we saw before? Or is this, yeah. Wow. Look at that. That'd be tricky, wouldn't it? Wow. Oh.
Is that the big one, or is that one of the softer ones?
The softer one.
Softer.
I'm wondering if there was just a small offset. If you look at this map, see how it's like right off? I'm wondering if it was actually on that hump. But anyway.
I'm gonna do whatever this guy says. He wants to go look at the hump. We'll go look at the hump.
What do you think, Anna? Right there. I think she wanted to stay on the transect, fortunately.
Staying on transect. Oh, okay. Have fun. Some interesting terrain for sure, man.
Yeah, it's very cool.
The current seems like a little easier, but just watch it, man. There's a lot to get run into, a lot to get on.
So it seems now that we have deviated, so 3 is closer than 2, so might as well head on to 3 and then come back to the level.
Okay.
We're also kind of, this dive is getting, like we're running out of time.
Okay. Great. So actually, if we can just cut across to 3 first. Skip 2 for now, just because we've deviated a bit. I guess maybe that hump's on the way anyway, so we can check it out while we go. But yeah, I think we'll go 3, and then if we have time, go back to 2 after.
Hold.
Yeah, see how it goes.
Sure. And just when you go downhill, if it's too steep, oh, there you go. You go downhill and it's too steep, you might have to kind of crab or even turn and go backwards a little bit just to see where you're going. And if it gets too bad, you might just have to skip a part and go ahead. But just check it before you do that.
Backward north, so we're going.
You guys have to just go for it. Serious chat.
So we're taking a little shortcut across pretty steep area of this small mount run. No.
Let's stick to the plan because two had a bigger plan, a bigger
Okay.
signal, and it's only 100 meters further.
Okay.
And we're kind of cutting across.
Okay, so yeah, actually we're going to 2. Sorry for the change up. Yeah. I'm sorry. I'm just I'm taking orders.
Taking orders.
Yeah.
Great, Jarvie. Yeah, so we have a change of plan here. So we're gonna head back to 2, continue heading to 2. And I put in a new waypoint also. Yeah, these scientists and they I didn't say that.
Yep. Flying by the seat of our pants.
Just seeing microbial ants, they would be like, oh my god. And you guys like, whatever. I mean, not whatever, but.
Ooh, big skate, it looks like here. Just hanging out on the bottom. Can you get a picture of that, please? These larger predators that we've been seeing, like chimeras and skates, are also an indicator that there is a lot of life here, right? Because you need a lot of biodiversity to support larger predators like those. Okay. Yeah, so I mean, we can just go directly to 2. Yeah, whatever's easiest for you guys. Yeah, let's go.
So between here and 2, you'll have to kind of go down and then come back up.
So another reason that we are re-exploring this site that's been visited before is that these seeps actually don't last forever. There have been cases in the past where scientists have gone to revisit hydrothermal vents and they've just shut down naturally, and those ecosystems associated with them have disappeared because they depend on the strange chemistry of places like this. So it was very exciting to see that that was not the case this time for us, and that the seep is very much still active with a pretty vibrant ecosystem around it as well. No worries. Got another one of these fish here. Looks like maybe one of those halosaur or something similar, which we've seen a lot of at all kinds of different sites so far. So in addition to collecting specimens and samples, one of the things we're doing on this dive is deploying some materials for an experiment on colonization in the deep sea. So hopefully we're going to be able to find a seep where we can leave some bones and some wood to study exactly how different organisms colonize these materials at seep habitats. Another skate down here on the left. And another skate here too. So you might be noticing that despite this rocky area, we're not seeing the same corals and sponges attached to the rocks as we were seeing the other day when we were on a rocky area as well that was non-methane seep. This is probably because those species just are not adapted to this habitat where it's very anoxic and there's, I think, different types of toxicity as well the closer you get to the methane seeps. What we have found are some possible carnivorous sponges earlier today. Those are really interesting because, well, you know, it wasn't until I think the late 90s that carnivorous sponges were discovered in the first place. They were discovered by a man named Jean Vassillet, who was actually scuba diving in caves of the Mediterranean on the coast of France. And he found that in these submarine caves where there's no light whatsoever, he was actually finding deep sea species that had never been known to be in shallower waters before, just because these environments really do mimic the type of environments that you see in the deep sea. And so what he found by being able to scuba dive repeatedly and with more time to these different submarine caves, he actually noticed that instead of filtering particles from the water like most sponges do, these sponges were actually catching zooplankton and absorbing them into their tissue, making them carnivorous rather than filter feeders. So very interesting story there. Huh? Oh, yeah. Can we do lasers, please? Is that a clam or a rock? That's a rock. Yeah, can we zoom in on these rocks? Oh, that's also something big over there, isn't it? Yeah. cuneiform floating by. You can kind of see that this discoloration on the floor is probably from some kind of microbial mat, which is also pretty common at seep habitats. Got a pretty large fish here. Does not seem too bothered by our ROV, this fish. Still slowly making our way to waypoint 2, which we hope has a pretty good sized plume from the methane seep, but we'll see how it looks when we get there. Got some kind of fish in the distance. That's another chimera for sure. Wow. You can really see how slowly even these big predators move. Pretty much everything down here moves pretty slowly. Yeah, and if you want to, for scale, these lasers are 10 centimeters apart, so this is a pretty long, pretty large chimera. Oh, there he goes. Oh, just swam right into a rock. Hopefully we don't do the same. Chimeras are actually, along with skates, rays, and sharks, I think the Chondrichthyes is the name of the class. Yep. And so they're pretty closely related to sharks. And you know, the common thing being that skates, rays, sharks, and chimeras all have cartilaginous skeletons as opposed to teleosts, which are bony fish. So one of the things that was so exciting about finding the ray fall a few days ago is that it's not only was it the second time ever that was seen, as far as we know, but I think it's pretty uncommon to see a cartilaginous skeleton on the seafloor. And we've got some more small corals here on the rock. They're eel or fish. Oh wow, big coral here. Can we just zoom on that coral for a sec? Do we want to snip, Anna?
No, we're not selling.
No.
We're not spectators.
Okay.
But we can take this one.
Yeah, so no sample for this one. We're just gonna try to zoom and get some pictures. It's very. What's that? Oh wow, we got some. Looks like a big remnant of coral, possibly still alive here. Can we zoom on that for a sec too? Yeah, or maybe those are tubes from worms. Wow. I think that's probably good. Thanks. Ooh, another huge fish. Yeah, so I guess we'll keep moving on to waypoint 2. It's more big coral, left and right. Ooh, wow. Skate and some more huge coral. We can keep moving, but if we just maybe fly by that coral a little bit closer if we can.
We're almost at one, two. It's
Oh, are we?
probably one, two ahead of us.
Okay.
Just beyond.
Yeah. Yeah, I guess we could just keep moving on.
You want to get a close up of that? Okay.
I think it's okay.
Carry on.
Yeah. So we're moving in on one of our waypoints, which we think has a pretty large plume from the methane seep. You can kind of see the landscape and the fauna changing as we get closer. These big corals. Wow.
Update to waypoint 13.
Some big squat lobsters. Okay. Oh wow. What's that?
Trash bag.
I don't, can you zoom on that for a second? Oh yeah. Oh yeah, it is. Never mind. Don't zoom.
Don't zoom.
Just a trash bag. Looked like something else to me for a second.
Probably an inside.
Yeah, it's a pretty suspicious looking trash bag.
Tied very well.
Yeah.
It's a long way to go to throw your garbage out.
I wonder what's in it.
I'm ready when you.
You're not interested? Just have a little look.
That's okay. Okay, so I'm going to pass you all off to Rachel Lauer, one of our rock and heat experts. So enjoy.
Yikes. Hello.
Okay.
Yes. So we're heading towards the seep target.
Yeah, that is appalling. Hi, everybody. This is Rachel Lauer from the University of Calgary. I am more of a geophysicist and a hydrogeologist than a biologist, so we are on a quest for bubbles today. We did see bubbles in the multi-beam data that were collected over this site. That's collected with a swath, so it's hard to pinpoint exactly where those bubbles are coming from. And they show up because bubbles have basically the density of air, and they are within water. So that density contrast allows us to image them in the water column. Other places I've been bubble hunting are much shallower, so I would expect the bubbles to be a bit easier to spot in those environments. These bubbles may be very small streams, small bubbles, but we're looking for something vertical that is then just coming up out of the seafloor and bending within the current itself. And what'd you see? Oh, lots of worms. So many worms. Okay. And clams and all the things that you find where there's lots of methane. Cool. Yeah. So if we can, it's possible to like zoom in closer if anybody does see something that might be bubbles, but we all keep our eyes out. I'm not trying to just. What's that? Okay, got it. Okay, so the darker colors are indicating mussels that are currently alive versus the massive graveyard of mussels that are covering this whole area. And it looks like a seamount, but it's not. It's all chemosynthetically formed carbonates. Whoa, okay. So this area was visited back in 2014 on another cruise. They also found lots of bubble locations based on the geophysical data from the ship, the multi-beam data, and actually touched down and visited a couple of those with Nautilus.
I want to deploy the experiments here.
Right here?
Yeah, because there's all these living mussels.
Yep, the living mussels. Okay, so it sounds like we're gonna unload some experiments on the area where we have the thriving mussels. Yeah.
There could be something incredible.
Yeah, like literally, we're in this area that we actually look. That's where we're not even there yet.
So we're gonna keep looking for.
Okay, my base looks like a seep.
All right. So the ideal place for these experiments would be an active seep. So we're going to keep looking to see if we can actually find those bubble streams that were imaged in the multi-beam. Yeah. Yeah, let's at least get to the, you know. Okay.
There's maybe some bacteria on each one.
Yeah. It's a white crab. That's a really good descriptor. Yeah, all of the stuff that we're seeing here are communities that thrive under cold seep conditions, so methane seeps. And the giant boulders that we're seeing are also formed by chemical processes that are the result of those seeps. So we're on an ancient seep and an active seep based on the data that we obtained earlier today. Now the problem is just seeing if we can find those bubbles, and if not, we'll go back to where we just were with the mussel community, active mussel community. Wow. Okay.
I'm not sure if this is your goal, but if you wanted to have a better chance of seeing bubbles,
Yeah.
having them come off bottom like 20 meters, so that
Yeah.
they move to back, and then doing a slow sweep and seeing in the summer. But also, if you're not looking for bubbles, we're just looking for seeps.
Yeah, I mean the bubbles are the seeps. So, problem is under this pressure, the bubbles are going to be super small. Yeah, I know. That's the problem.
Yeah.
I'm afraid as they get higher, they will get bigger. Tend to. But,
Yeah, no.
but it's also like, where did we see them? Was it right around here or at point 2?
Those are here.
Yeah.
But I wouldn't go super close to that accuracy, just 'cause
Yeah,
I
yeah.
also,
Right.
like I grabbed the wrong thing.
Yep.
But, yeah. I mean,
And we can also go up and zoom in as well, but also pan around.
yeah, I mean, it's up to you guys for your goal. Like, if you want to
There's
see
like a
bubbles,
mound here.
it's best to pop up. Yeah.
Yeah.
And you'll be able to see them,
Yeah.
especially if we can see them here.
Yeah. Do you see anything in the sonar now?
No.
No? Okay. Okay.
But that doesn't mean we shouldn't check it out. I'm just saying.
Yeah.
Yeah.
Oh, that's cool.
It could be there's terrain kind of
Yeah.
higher, and we
Yep.
wouldn't see it then.
Right. What about heading to that higher spot and taking a look around as suggested? Yeah.
It's a great point, Rich.
Go back to your map.
Hmm. Yeah, so this whole landscape is basically the process of bacteria oxidizing methane or sulfide for energy. And the byproduct of that, oh, another skate. Is that the same one? Yeah. It is? Just keeping an eye on us? Okay.
I'm just going back to the ship to catch us not too far away.
Yep, the ship's too far. Yeah, so instead of photosynthesis, they are using chemicals for energy. Not today, but tomorrow we hope to be visiting a similar site. Yeah, it's the same skate. It's following us. Yeah, I just learned earlier today that the tube worms that also are indicators of methane seeps can also live for centuries, apparently. Did not know that. And in a lot of these exploration mode dives, we're actually using biology to identify the seeps, because the biology is pointing us to the locations where they're, in particular, using that methane to create their habitats. So the tube worms and the mussels are indicators of active seeps, and the dead remnants of them are indicators of past seeps. And these are more persistent than like vent sites. Hydrothermal vents can turn on and off within the same week. These tend to be more persistent over time, so you get these massive formations like the ones we're seeing. All right, so there's some, should we, on the active seep here? Okay, yep. All right, Charisse, do you want to, there's some living mussels as a potential target? Yeah, I think, right? That's what.
Yeah, and the bubbles in it.
Yeah.
Sounds good. Thank you. Hello, guys. Hello, everyone. So we're actually going to deploy the second half of our experiment. If you were online earlier, you would have seen that we were deploying on the periphery of these seeps. So now that we're in the active zone, we're going to deploy the cow bones and the wood fossils as well. So we're trying to get an area with a lot of live mussels, and we're going to deploy that. Okay, similar to how we did earlier. Yeah, I know, but I have to say it. Anywhere there? It is a massive curve. And it will feed on the bones. So yeah, wherever's best for you to land. I'm not entirely sure.
Pretty steep.
It is.
Yep.
But it will try, if we can just try to get a patch with like live mussels, that'd be good. Yeah, so these colonization experiments mimic what naturally happens. Yeah. Naturally mimic what happens typically in natural world. So when wood falls in the bottom of the ocean, it creates a biological hotspot for organisms that you don't typically see anywhere else. We don't actually know where they are the rest of the time, but they are found specifically on wood falls. And similarly, when whales fall, die and fall to the bottom of the ocean, it happens with those as well, with bone boring worms. So the cow bones will be mimicking those kind of whale falls, that whale fall activity, and the wood falls that would naturally happen, we're doing with the wood parcels that we are also going to deploy. And then we'll see what colonizes in our waters naturally.
Is this okay, or do you want to go higher?
Can we get like a bigger one? That crab is weak. Just because like when we space it out, I want as many alive. All
This
the
is super steep though. These cows here are going to come tumbling down this hill.
Okay, so do you suggest down there then?
You need a little bit flatter, else you're going to lose them when you drop them.
Okay, so
Yeah.
okay.
We'll start at the bottom, eh?
It is steep, I see it now.
It's kind of,
Yeah.
they've gone now.
it have to be that first natural reconstruction? Okay.
Maybe higher up is flat.
Yeah, if we could choose. Yeah, that's one? Yeah, I was thinking. Very nice. That's very nice. Because I was like, it seems bordered, so in case of anything, it shouldn't tumble down. Yeah, that looks pretty good. That looks perfect. It's not gonna work. 164 on the nearby, it will move to the thing. So it's not too, if it's slightly off from it, it's up. There's a lot of muscles. A lot of live muscles. Amazing. Look at that. Look at those crabs. Stunning. Wow. And you can see some bacterial mats around as well. Do you have enough room to send out the porch? Okay. Alright, so just like we did this morning, we're gonna set out the bones and the wood. We set them up basically soil all just intersperse them a bit, and then we'll do the side marking.
Yeah. Anyone you want to start with first? Spoons or what?
No,
Doesn't matter.
not too much. Whichever is easiest.
We have to watch out.
Okay, let's watch out.
All right, so we're going to begin. As you can see, we have a very healthy area of mussels, living mussels, which we believe will be some of the things to colonize the wood, especially.
Do you want any imagery first of this?
Can you just grab some frame grabs of it, and then when I start deploying, I'll tell you which one to put down, and you can put them for the experimental. Yeah. And then we'll take a larger grab of the entire experimental set to put it down as well.
Stop already.
So you can grab frame grabs all now. Been doing it. Great. So we can begin. Yeah. Sean is on it.
Okay.
Torch coming out.
Torch coming up. You're going to grab the two off the top first?
Alright, see if we can see the number on that when it comes out. Wood.
Nice. See if we can see a number cord as well.
1. Wood 1.
So that's the experiment ID.
BNW1. Yeah. The crap. Perfect. BNW1. This one is 5. This one coming up now will be 5.
Did you see it better when you landed? Did you see underneath? When you were looking for the numbers? You like bent down to see if you could see around corners.
Okay. Right, so that was 5.
Okay, 5 box. Box open.
And these are all wood so far. Yeah? Two? Yes. Okay. After this one, we'll take a pause, just so they can be sure.
So you're making these the logs for sure.
That's
for each
what
one.
they did, yeah.
Yeah.
Before.
Okay.
Yes. Okay. So the first one is the 1, 5, and 2. Bringing up number 4. Can we disperse them a little bit more?
If you put that close to the porch, you can.
So you have to watch out. Huh?
Am I not pushed out on the left?
No.
Do you need anything in the back?
It's on the side.
Yeah, these bones right there.
Hard if you don't watch out properly. I could always take the other arm
Rearrange.
and move this.
Do the fishes samples? This is what they're coming out.
Full check? Okay.
Yeah, so if you want to dump them in the bottle, but still get rid of this first.
He's gonna use the full arms, right?
Okay, got it. Look at that foot just there. Same.
Good porch coming in.
Is that all five? Should be. 1, 2, 3, 4, 5, correct. Sorry, what number was it?
Two.
2, yep. Yeah, so we have to intersperse the wood and the bones together so we can have all bones in one area, all wood in one area. Just wood and wood.
You don't want it like you did it the first time?
It was like that the first time. There was wood and bone interspersed. We don't want all wood in one area and all bone in one area.
He'll disperse it now.
Okay, just that.
So I'm gonna read that now.
So I was just making sure we don't put only bones in one corner.
No, it has wood on this corner. Don't worry, don't worry.
So if you put So if you put a wood over on that side, and yeah, and a foot over there, that'd be good.
Yes.
I just double check. Like the crabs are coming in. I don't know. 5. Carbon 5. Yes. Okay.
Close. What do you want to rearrange? Anything?
No, we just need to put them line them up.
Okay.
Yeah. Start the box.
Yeah, there should be a monkey switch with the green.
You want to switch them up?
I think that's better too.
You happy how it's arranged, or you want to swap anything?
1 second. Let me just double check. I am good.
Sure. Real up, side real.
So when we deploy
Side reals.
It's all the way out, isn't it?
No?
so no need. Okay.
It's good.
Yes, perfect. Look at that monkey juice. All right, can you take one waypoint? And then we'll zoom out
Not that. See if we can-
with the lasers on as well. And we'll grab a frame grab. Is that just any words? Okay, site 3 active. This is great words. The site is 3. I'm good again, I'm good. Okay. Waypoint done? Great. And just,
Come up.
yep. And we'll take the frame. Shana, when we're up and lasers are on it, we'll take the frame down. Thank you. All right, thank you to our pilots for setting that up so nicely. Now we have our deployments for the next year or so on this active site, and we will be able to collect it hopefully in a future expedition and see what has colonized it and fed on it. So what we're gonna do is just zoom out, fly up and zoom out a bit. And there's a lot of giggles happening in Mission Control right now. But yeah, we'll take a frame grab of the entire setup so we have it with lasers on.
I think the lid should be on the upper, no? On the, like the lid should be floating, no?
No, we knew it was saying, so that's why the monkey's fist is up with the reflective tape.
Second we turn around.
When it settles. Is it wrong? Oh, I see them now. Okay. Those crabs are going in immediately, I can tell. Motion, please. Thank you. See everything? Do I see everything? And grab it. I think we're all good. Thank you so much, guys. And I'm handing back over to Rachel now.
Man, look at those crabs. They're all lining up for a feast. Okay, so we've just deployed the experiments. Hopefully in a year, we can come back and see how they did. In the meantime, we're just gonna fly over this amazing structure that is massive amounts of carbonate. They're basically forming these mountains at the seafloor over millions of years, so 5 to 30 million years. are the estimates from the paper that sort of focused on this area. Yeah, this whole thing is encrusted in worms and mussels and not sure what else we're looking at. Oh, we're checking out the experiments. I'm surprised those crabs haven't moved in yet, but another crab. Yeah, so this whole formation here and same location tomorrow are all the result of huge amounts of pressure and biodegradation at greater depths where there's higher temperatures lead to hydrocarbon development, and one of the things that occurs coincidentally with hydrocarbons is methane. And over millions of years, these enormous amounts of sediment that are scraped off of the Atlantic plate that's scraping it onto the Caribbean plate has formed this massive mound of sediment. And at the base of that, you have lots of hydrocarbons that are combining with all of this methane to kind of make their way to the seafloor along these faults. And, oh, another one, another skate. So this whole region is covered in seeps that lead to the development of these types of structures that provide amazing habitat for not just the mussels, but also we saw earlier all of the inactive seeps that are ancient seep sites are now providing habitat for different types of creatures. So when there's nothing but a muddy desert, any of this hard substrate is a nice refuge for these critters. On the right, is it? Making a tiny adjustment. There's another coral. Is that a black coral? Does anybody know? Come on, biologists. I don't know. Oh, yeah. I think so. Yeah, don't open that one.
Front one? Why not?
No, the octo's in 1A, the bottom. Yeah, we're good. So over time, these sites are migrating. So some of those faults that are providing pathways for that methane and hydrogen sulfide and other chemicals to make their way to the seafloor can open and close as a function of many different things. An earthquake can clamp down a fault and make it no longer permeable, or you can just have the carbonate itself sealing off a fault, and it migrates to find another place where it can seep at the seafloor. So these are not like static in geologic time, but they are in the scale of our lifetimes. So that's sort of the big difference between, well, one of the differences between cold seeps and vents. Vents can turn on and off, as I said previously, in like over the course of a month. And in fact, we have instrumented vents in other parts of our oceans and expected to see them flowing the same way they were a year later. And when we collected our data, it turns out that every couple months it would turn on and turn off. These are on a longer time scale, but geologic time scales, you know, this was probably started being active 30 million years ago. And so over that time, the active parts of the seep have migrated and added to this calcium carbonate that we're seeing. So we're heading to which one now?
I'll just go back to 2 and then we'll get closer to move and we'll start heading into
Cool.
Yes.
Awesome. Whoa. The ship's moving.
Are you going back here? No.
Is that looking up on those things?
What's that?
Has that been on there the entire time?
Yeah? I thought you were going down there.
Have we? I've not got that here.
What's that?
What can I do? So I was looking at bubbles.
Oh,
Didn't see that.
yeah. Well, that's too bad. Yeah. How far away is that? Cool. So we're still just exploring this area. Parts of this have been explored, I guess, in 2014, and paper focused on this area and another mound to the west of us. But as you can imagine, in 12 hours, you can only do so much, and probably exploring like, you know, less than 2% of this structure that we're on. That is another coral that I'm not going to be able to identify, but someone will make fun of me for later.
zoom in on that giant mat of
Great.
corals again. Oh, beautiful. Is that the one we got a sample? That coral? No. So tonight, after we explore these two sites, we'd like to get onto the muddy area so we can take a heat flow measurement. If we have time, of course. But one of the other objectives of the next couple weeks is to examine another fluid flow formed structure at the seafloor, and these are mud volcanoes. So this part of the accretionary prism is covered in lots of seeps, as well as areas where fluidized mud from up to 10 kilometers below the seafloor are pressurized and mobilized to flow from the base of the subducting plate all the way up to the seafloor and form these structures that have been mapped by oil and gas and are associated with hydrocarbons as well. And in some cases, those those mud volcanoes are moving fluids from 10 kilometers below all the way up to the seafloor. And samples taken from them suggest that, yeah, these are probably sourced as deep as 10 or 11 kilometers at the base of these sediments. So that plumbing is providing a pathway for warmer, deeper fluids to make it up to the seafloor. Another black coral. And the other thing that's making up a lot of these rocks are the same communities that form when an active seep is present. So a lot of the mussels and tube worms and other things become that fossilized limestone. And I've mentioned this before, but the reason that limestone looks different than a lot of terrestrial limestone is because it is sourced adjacent to and within hydrocarbons, so it takes on a much different color than lighter limestones that you may see that on land. Whoa. Okay. That's steep. Yeah. Why not? That is a steep slope.
Sure.
Or you take over.
No, you do it. Let's do
come
it
on.
together.
Tag teaming.
No, let's do it together.
All right.
Okay? I'm good. I'm gonna stand. I'm
Are you
gonna sit.
sure?
Yes. Yeah.
All right. Well, I am honored to introduce our next person, Shirley Pomponi, who is a sponge specialist, sponge hero, super sponge
We're
person.
fangirling.
Yeah.
So this is gonna be the first time we do a duo
Yeah.
dive lead, because I know very little about methane gas seeps, and Rachel and her colleague Rob Perrin have been spending every night mapping the seafloor bottom and looking for bubbles, and they found them, and I want to make sure she's here when we get to that area. So anyway, as Rachel said, I'm Shirley Pomponi. I'm from Florida. Worked most of my career at Harbor Branch Oceanographic Institute and Florida Atlantic University in Florida, and I focus on sponges, sponge biology, and sponge biotechnology. A little while ago, we collected a sponge that is a carnivorous sponge. So it, instead of filtering out food from the water, it has adapted a different way to eat. It catches crustaceans, tiny shrimp, on its surface with specialized microscopic skeletal elements called spicules, and the surface of the sponges is like Velcro, so the food gets trapped there, and then the sponges mobilize cells to completely engulf their prey, and then over, probably days, they digest that food item and usually spit out the chitinous exoskeleton. But these sponges also may take advantage of the fact that they've got bacteria living in them that are called chemoautotrophic. So they get their food from the chemicals that are in these seeps. And there's some strong evidence to suggest that maybe the sponges are using some of the energy and food that's produced by these microbial symbionts that live within the sponge and that are using the chemicals for their energy. They may use some of that for their nutrition. So these sponges may not only be carnivorous, but also chemotrophic, chemosynthetic. So how'd you find the bubbles?
Well, basically this ship is equipped with some of the best equipment for mapping the seafloor at any depth. We have 3 different multi-beams that are operating at different frequencies that allow us to map at high resolution shallower areas surrounding the continent, a little bit deeper as we get off the shelf, and now we're into yesterday we were into areas that are even deeper than 2,000 meters. So we use a multi-beam to look for tiny plumes in the water column that show up as contrasts between the density of water and the density of gas. So it shows up as a little plume.
So the multi-beam is just like sonar, like lots
Yeah.
of sonar pings
Yeah,
that are
so
going
it's-
down, and then
it's-
you're measuring like how long it takes to get from the ship to the sea floor and back up to the ship, and that tells you something about the depth, and
That's right.
it'll tell you something about what kind of substrate it is, if it's hard or.
Yeah, so along with that, we also have another instrument that's called a sub-bottom profiler. And that is basically capable of doing these sort of mini seismic surveys, which are also sending energy from the ship down and receiving that energy back. And if it's soft, we can get penetration into that sediment that tells us something about the structure of what's happening, maybe how deep the soft sediment is. And in some cases, it can show us where gas pockets are, so indicating potential seep locations. And here, we may not see the seeps, but we see the evidence of the seeps in the form of all these critters, these mussels, and as you mentioned, there's symbiotic relationships between the microbes and the mussels and the tube worms, and all these creatures are creating habitat for things like sponges.
So how deep does the sub-bottom profiler go?
Well, it depends on the substrate. So if it's like a clay or really high porosity muddy stuff, which is a lot of what we've seen at our previous dives, you can get penetration all the way up like 100 meters below the
Oh
seafloor.
my gosh.
But if it's harder and more consolidated, you're only getting, maybe tens of meters of penetration.
Okay, so I have a question. So these bubbles come out.
Okay to start heading up to waypoint 3?
Yes.
Or you guys want to?
We're good. That sounds good.
Yeah, that sounds, yeah, let's do that. So when these bubbles come out, just like any gas, as they rise to the surface, the bubbles are getting bigger and bigger, and then do they break up into smaller bubbles and then make this screen? And is that why the sonar the multi-beam can detect them?
Yeah, so they are getting bigger as they get close to the surface. They don't make it all the way to the surface from the deep ocean.
New waypoint bridge.
But yeah, the way they're detected is through this sweep, this energy that's sent from the ship along a swath. As that energy is received, we can actually see these little
Blips.
anomalies.
Yeah, can they see
basically.
that image that's up, that we're looking at? I wonder.
I don't know.
Let's see. Alex,
Yeah.
can you show them that, the map that shows the bubbles, please?
Yeah.
Okay, thank you.
Yeah, so we're going to put a little insert into the dive so you can actually see how that information comes back to us and where we can try and identify regions of methane seeps.
We got super excited when we saw this because we've been searching for this. And of course, it's just been a couple weeks ago that there was that seismic activity, the
Oh, God.
earthquakes.
Yes.
Yeah, off Venezuela, and up into this area as well.
Yeah. Well, and the same fault that ruptured and caused that earthquake extends all the way east to the top of Trinidad. So that's a strike slip fault that separates South America from the Caribbean plate, and that earthquake that happened to the west of us has actually led to some seafloor changes in southeast Trinidad, which is wild because there was no earthquake here. But
you
all
got
of
that uplift.
yeah,
Yeah,
there's
that uplift.
lots. I think what is this? 20 feet of
20
uplift.
feet of uplift. Yeah.
Yeah.
On land. So what you're seeing there, all those like bluish purple dots, those are the bubble. That's a bubble stream, right?
Yep. Those are basically just anomalies in the water column that indicate a different density, and in this case, the contrast between gas and water.
So these animals we've been seeing here are ones that are really indicative of these methane seep areas.
Yeah, and a
Or
lot of
gas
these
seeps, yeah.
giant boulders that we're seeing are also just old mussel colonies that have been sedimented.
Oh, really?
And yeah, so the formation of the carbonate is not just forming carbonate, it's also encompassing a lot of the structures that are literally old dead colonies of animals that were thriving when it was active and are now providing a substrate for or a
other
platform
other
for
things to colonize, yeah.
exactly.
Right, RV. Yeah, I have a waypoint to the north of you. I think you're heading south.
I think they're moving the ship.
Okay.
So we're going to 3 now, is that right?
Yeah, I
Okay.
think we're moving over to
Cool.
moving to waypoint 3.
And so we didn't feel any earthquakes, or there were no earthquakes in Trinidad,
yeah.
at that time. Apparently there was one today, like a 4.2
really?
or something. Yeah,
Oh.
in Trinidad. Obviously we didn't feel it. But that much slip at that shallow depth to the west of us can actually lead to deformation this far to the east. So there are hundreds of kilometers to the east of where that rupture happened, which is wild to think about. So what was submarine or below water is now above water in this region in the southeast Trinidad. And it's been in the news, but I don't think it's gotten a lot of
Press.
a lot of press or interest in terms of geology. Geologists. Oh, there's another skate. Oh, we're way up. I see.
And those whites are the white is what is that white? Oh, that's that sponge.
Yeah.
I think
Is it?
Diva
Are
said
those
she-
sponges?
I think so, but you know what? We tried collect. I
Yeah.
know Diva collected those a few years ago when they had a cruise here,
Yeah.
and they just kind of
Crumbled?
Yeah, fall apart.
Okay.
If we get to an area where there are, you know, there's a large amount, and we might try and suction them up. I remember she sent them to me a few years ago to try and
Really?
identify, but they were just mush.
Oh.
And then are they? Oh, and the bacterial mats. We see
Yeah.
that white.
Those
yeah.
look like bacterial mats. And another, is that a coral? Yep.
Yeah.
And a brittle star or something wrapped around it.
There's maybe a squat lobster. There's another
And there's another
mussels.
colony of mussels,
Mussels.
and those appear to be alive, so maybe indication of another active seep.
I hope so.
Yeah.
This is not far from where we put the cow's food up.
Cow's foot. Should we head to that? Away from it then? Where's our next waypoint? Off, is it off the mounds? Oh, I see. We didn't go to 3 yet. Yep. But we're heading there. So 3
This
is
is
on
really
to the next
different now,
ridge,
yes.
and then 4's off.
Oh, okay, so we're going to go up to 3 and then back over
Yep,
to
and
4. We're
then-
not going to continue along. No.
I think to
To
waypoint
set-
3 and then down. There's like 2 structures here. There's I think they're cleverly named Mound 1 and Mound 2, and
At waypoint 3.
yeah, no, well, we're still on the same mound.
Yeah.
But we're gonna go down,
Down to the
into
okay.
and
all right,
maybe
cool.
do some heat flow if we have time.
Oh. Oh.
Yeah.
So that's your specialty.
Yes. Yes.
One of them.
Oh, another giant colony of mussels. Living mussels.
Some alive, some dead. See the dead
Yep.
ones, all these dead shells, the white ones.
Yeah, so just to answer some of the questions in the chat, with a disclaimer, not a geochemist, but these bacteria that actually live at these seep sites are oxidizing the methane or the sulfide that's part of the fluid that's coming out for energy. And one of the byproducts of many different reactions is the formation of calcium carbonate, which is what's forming these crusts. A lot of the shells that these animals have that are also inhabiting this area are made up of the same material. So what you get is a platform that's made up of chemical reactions as well as biology that are forming this giant platform. Yeah, so there's organics as well as chemical byproducts of these sort of symbiotic reactions between the microbes, creating a place for tube worms and the mussels themselves. And then we've also got, I think, are those sponges or corals?
I can't. Maybe we can get a close up when we get in position to do that and get a close up of the little white things.
But then the tube worms are also hosting bacteria.
Right.
And
that's where they get their energy.
That's right. So they're also getting energy from these bacteria. So, and I did not until earlier today know that tube worms were some of the longest living animals on this planet, and they can live up to like 250 years. That is, what is that? I don't know. I mean, you can see are those dead tube worms and mussels. Yeah. So again, this is, some of this rock that we're looking at is, or a big part of this is biologic. You can't really do geology without understanding chemistry and biology. So.
So those kind of white, creepy, branchy things are sponges. For
they
sure.
are?
Yeah,
Cool.
These white, creepy, branchy things are sponges. The other, like fan-shaped things, are some kind of coral. I don't know if they're a hydrocoral.
They are the sponge.
What are they? The white.
Yeah.
These, the white brick.
Yeah.
I don't think so. I think the branching ones, these little kind of more creeping, branching ones, definitely are sponges.
But do we know the type? No.
The other ones, the ones that look like seagulls, are not. They're not. You're welcome. So I mentioned yesterday, I think it was yesterday, that the growth rates of some of these same types of tube worms that live in the Gulf of Mexico were measured. The growth rates were measured by Dr. Chuck Fisher many years ago using the Johnson Sea-Link human-occupied vehicles, manned submersibles. And the engineers at Harbor Branch at my institute developed a chamber that they could deploy and put kind of a plexiglass chamber that they could put over top of the tube worms and inject a dye that didn't kill the tube worms, but it stained the tube. And then they could go back like a year later or 6 months later, whenever they got the opportunity to go out again, and they could, first they took the chamber off. So the chamber was just used to put to stain the tubes. But then they could go back at certain time points and measure how much of that how much that tube had grown, and so you could get an estimate of how old the tube worms were. And I'm not sure if individual worms live that long, but I mean certainly those mats can be really, really old. So we got a question about whether or not the oxygen saturation would decrease near the methane seeps. What do you think, Rachel?
Oh, I think that's a question for a chemist.
Well, we'll be able to monitor that actually, right?
Yep.
Because they've got that on your live stream feeds. You'll be able to see oxygen
Can they
saturation.
see that? Yeah, maybe. Another thing that does happen as our oceans warm up is this methane has a very specific stability range, temperature and pressure, and a lot of continental shelves where historically these materials are stable are now forming or degrading to the point where we have active methane seeps in places where we didn't previously.
Coming up on another
Oh wow,
cluster
giant
of
mussel
mussels,
bed.
Yeah.
Cool. So probably indicating another active seep here.
Yep.
Do you want me to go get dinner and then come back?
You can go.
Yeah.
Yeah, go ahead.
You sure?
Yeah. Yeah, go ahead. We'll yell if we see some bubbles coming up.
Yeah. Well, I'll stick
Or you can
around.
stay.
I'm not.
You can stay. Or you
yeah.
can just ask somebody to make a plate up for you. That's what I did.
It does feel like we eat every 5 minutes on the ship.
No, I'm kind of hungry right now.
Yeah, you can go too. Yeah, these microbial mats are insane also. I can't see where we are on that one. Yep, yeah. When we see those microbial mats, be cool to zoom in and see any snails or other critters that are munching on that. Anna, do you see any? We have. Yeah, if we can zoom in a little bit on that.
Anna, do we want to collect any of those stylasts? No.
Tomorrow. We're not making any collections.
No
today.
collections, all
Tomorrow
right.
we will.
No collections.
sure.
Tomorrow. My little friends Josephine and Reagan are watching from Indianapolis.
Nice.
Back to position yourself so you want to trust back
I
yourself.
guess that's what those
forward.
dots are.
If you want to pitch
That's
white dots that we see on the. So this is a mat of dead tuberous, right? It's stuck on it.
yep,
So I think the white dots are just feeding on the mat.
on the mat
Raised
itself.
just
Yep.
slowly.
There's like layers and layers of ecosystems.
Hannah and Maya, hi to you in Massachusetts. We've got a lot of our next generation of scientists
That's
watching.
right. I hope we do.
Yep,
We need more of us.
yep.
Get them young.
We did get a request for jellyfish, though.
Wild. What is
You want to go eat them and yes.
What's for dinner?
Curry.
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 Nice.
Three different kinds. 3 different kinds of curry for dinner.
Nice. No shortage of
And
curry.
rice, yeah.
Which coral?
I don't know.
Do you want me to put some food?
Megan already is.
Anything specific you want to see now or just keep moving?
I think keep moving towards the next
Yeah.
point. The objective for this cruise is exploration.
Yeah.
So we've seen a bunch of different parts of the seafloor, but I have to say this is the most exciting
yeah,
so
we're-
far.
Of course, we're prejudiced, of course. Can never get enough sea
No.
cucumbers.
Yeah, I mean, I like all the critters we've seen, but like actually seeing structure and
Yeah,
million-year-old seeps and
it's pretty cool, and all the different things that are living in this area too.
yeah, and how extensive it is. I mean,
Yeah,
this is like this is
yeah.
the size of a seamount, and it's just chemistry and biology, which is crazy. Oh, there's
Yeah,
a
there's a
cool. Look
coral.
at that coral.
Yeah,
Wow.
I know.
Whoa. Cool. Okay, let's chill out here for a second. Zoom in, maybe. What's on that? That's the little crab doing the.
Yeah, that's a.
Yeah. The peace sign. I mean, I know that's not what he's doing, but that's what
But
it
we
makes.
can say that.
We have sampled some corals, but we're
Yeah.
not sampling today. This is gorgeous.
Yeah, really pretty.
So this is a black coral, right? Oh, come on. Diva's, I can hear her yelling at me somewhere. Is this a black coral? What? Is it? How can we zoom in?
She's doubting.
I know.
She's doubting.
I'm not
Juliana.
doubting you. You're a biologist. Oh.
Oh, the
Okay,
bri-
there's a brittle star
stars.
wrapped around. Oh, it's beautiful. Ooh, can we zoom in on that little one down there? Okay, there we go. Wow. Caligorgia. Oh. So cool. And then there's that
There's a
benthic
squat-
ctenophore, which is something I learned earlier today.
What letter does that start
I didn't
with?
know ctenophores could be benthic, so yeah. Nice. Oh, this is beautiful. Can we see how big this is? Do we have lasers?
It's big.
I mean, I
It's
know it's
pretty
big,
big.
but I can't tell
yeah.
how big.
Well, we can tell. Get some lasers on it.
Oh, wow. Okay. Yeah, it's huge.
See, it's big.
Yeah, that is a big, big boy. Yes. Or girl. Yeah. Thank you.
Or each year are they?
Over a century old.
Really?
Yeah. Yeah. So cool.
Yeah, we're thinking that the little white ones are another kind of coral, stylosterids or stylastids.
On the left there?
All of
yeah.
these in here.
Okay.
Yep.
Wow. Is that from us or
Yes.
the current? Okay.
No, that's from us.
Wow.
I know.
Hold on, crab.
Yes. That's a
Okay.
little squat lobster. Doing the peace sign.
Is it? That's a
Mhm.
squat lobster.
Mhm.
Okay. Um. I'll come up in 15.
That's beautiful. Just gonna see if we can start doing
That's beautiful.
Mm-hmm.
Marco.
Really close up.
Yeah.
Nice.
Now, how are you supposed to count? 8. How does... Tiny, tiny. Oh, look at that brittle star chilling out, waving its arms around, eating. What about these
What
little...
are those things that are called?
Yeah, those little red bumps.
Isopods.
Huh.
Amphipods.
Wow.
Amph-
Are those baby
amphipods,
brittle
are they
stars? No.
amphipods? What are they? Are they amphipods? Those little ones?
No, I don't know what it is.
Wait.
The
wait,
little,
wait,
the
are
little
these
red
here?
things.
No,
Yeah.
they're
What are they? Amphipods?
Yeah, I think they're amphipods.
Okay.
Yeah.
All right. Cool. It seems like if you just keep zooming, you find more
More
stuff.
and more stuff. Sponge.
Yeah, and you can see all the
Oh,
the animals that make up this rock on the right. That's cool.
all right. Shall we move on?
Cool.
Let's
I think
move
we
on.
can move on.
Let's move on.
As much as this is, we could zoom in
can
on this
spin
all
hours.
day. Yeah. Yeah, you can see the tube worms that are encrusted in that rock as well.
Mhm. It's amazing to see all this structure.
It's wild.
I know.
And you start realizing that this is just life on top of life on
yeah.
top of life over millions and millions of years that's creating this whole thing. Oh,
And all
what's
from
another
those,
one?
and all from the methane.
Yep. Yeah,
That's
this is
providing
all
energy. Yep.
life
These-
through chemistry.
Yep.
Yeah, this is, oh, there's another coral. Okay, another big one. In the back there. Is that? Yeah, can we zoom in on
one?
the closest one? Yeah.
Ah,
What are those? Is it dead or there's something alive on it?
well, the white things are the stylaster.
Oh, the stylaster
But
that you're
I'm
talking about.
not sure what the branch is.
Yeah.
It's dead, whatever it is.
Mm-hmm. Well, it's
But
gonna
it
it's
was-
gonna
but it-
provide
but it was
more
subs- but
rock.
it was substrate.
It's future future habitat.
Yeah. Okay. I think we can keep going.
Yep, we're good to keep going.
Want to see the big one as well?
Yeah, I mean, whoa. Okay. And do we know what that is? That's huge. Can we zoom in on? That's, yeah, what is that? No, another octocoral? Bamboo? Bamboo.
Oh, bamboo. Bamboo. I can't see the little lines in it.
Oh, look at all the little guys on it. Oh my God. Those are all, look at that. Those are all lobsters.
Geophysicists get really
Yeah, I know,
excited by
okay.
all this stuff.
Well,
When they see animals instead
I didn't
of
realize they're like
bubbles,
evenly spaced. Look at them all
they're
with their
like
arms out.
respecting each
Yeah.
other's territory.
They are, yeah. Arm's
Yeah,
length.
arm's length.
Yeah. Oh. That's cool. Wow. Okay. This is the place to be, obviously.
Well, it's very high.
Yeah, you're hanging out in the current, catching food. Wow. Yeah, exactly. Yep. Oh yeah. Yep. Can we turn off the lasers
Yeah.
and get some zoom ins on the good stuff? I'll go get some food.
Okay.
It's
Go
very
eat.
very fun. You join me in mine next
Okay,
time.
go
Yes,
eat.
please.
Okay. All right, my gal pal Rachel's gonna go eat. Thanks, Rach. All right, unfortunately, you're stuck with just me for the next 20-some minutes. You want more close-up images of this?
Sorry.
You want more close-up images of this, or should we just keep on going?
I think you were taking some frame grabs,
Oh,
like with the lasers, to
oh, okay,
then be able to calculate.
okay,
And then we use
okay, so you're taking frame grabs?
Been taking
You've been taking them, all right, so we good? We're good, we good to go, Robert? Robert, you wanna take some more?
yeah, we're good.
We good to go? Okay. Oh, that's pretty, isn't that? Oh, wow.
I've just been snapping them.
Okay, all right, sounds good. Okay. Oh, these are so pretty. So in addition to the video, so you can see those segments now. Somebody asked about not being able to see the segments for the bamboo coral, so you can see those segments in the stalks now. Yep. Beautiful. Polyps have now retracted their tentacles before we saw them extended. Yeah, you can see on the one of the other branches, some of them are still open up. Really nice. Beautiful. We're good. We can keep on, yeah, keep on going. Thank you.
Go ahead.
Hello. Could you start moving towards the waypoint? Can be slow.
Yeah, go right to start. I'll just go moving north towards the waypoint.
Yeah, that's good. Thank you.
Cool, thank you. So we're on our way to the next waypoint. Ship is repositioning, so we'll make sure that oh, is that another octopus? Yeah, looks like another octopus. So that the ROV can stand, directly underneath, not get pulled off. What do you want to do with the? You want more video of the? Or just take a little bit of video of it. Oh, nice, very cool. That's okay. We can keep on going. We have.
Let's do that.
We're good.
make some good close-ups.
Good close-ups of this, okay. 'Cause okay, so yeah, so if we can settle and get some good close-up video of this.
Stop the move, will do, thank you.
Yeah. Okay, that's good. We don't wanna- Yeah, that's good. Thank you.
Very good.
Yeah, go ahead.
Could you move the vessel again?
Okay, start moving 0.2 knots north.
So every now and then we're seeing some red animals that are attached to the rocks, and they're called benthic ctenophores. So a ctenophore is comb jelly. And we usually see them in the water, in the water column, kind of swimming in the water column. But there are some that are adapted to attach to something hard. So they're benthic, so they're attached, and they don't swim away. So they're called benthic ctenophores, comb jellies. Seeing the remains of another octaqual branch, and it's got stylaster corals and a squat lobster on it. Oh, there's another. I'm really surprised at the different types of corals that we're seeing, and so big too. Kind of looks like spaghetti, doesn't it? I think the plan is that we're going to be diving on another methane seep site tomorrow, so we'll get another opportunity to look at all these cool habitats and really diverse communities of animals living here. What is that?
Great, Jonathan.
Good. Hey, could you increase to 0.3 the speed, please?
Yep, 0.3.
Thank you.
we're at 1,255 meters. We're not seeing a big change in depth because we're running along the top of a ridge. Right? So we're starting to see some more bacterial mats now. White along the on the sediment. Yeah, let's just take a look, just get a closer look at that. If you can. Oh, you can. Yeah, that's right, keep going. You can it?
We're hitting the heat flow on the rocks.
So,
Yeah, so maybe keep it up or like keep the back end away from the wall a little bit.
yeah.
Yeah, there's a camera that's on it, but it's in the lab, the other lab. I don't know if we can put it on the screen.
Yeah.
plate somewhere. No. The thing over there, what go inside. Ah.
Oh, there it is. Yes.
Thank you.
What's that? No, the heat flow probe is hitting the rocks and then.
We'll give you this one in a minute.
Boy, if I didn't know better, I would say that looks a lot like those zoanthids that attach to the stalks of the glass sponges. But I don't know. Yeah. It looks a lot like zoanthids. Yep. Count those tentacles. More than 8, not an octocoral. Can we also take a look just past that? There's something white. Yeah, let's take a look at that. Okay. Shell. Dead clam. Mm-hmm. Megan, you ready? Yeah, you ready? Okay. Time for me to switch out. Thanks for spending the last hour with me. Yeah, I thought it was a sponge too. Anyway, Dr. Megan Conkling from Harbor Branch Oceanographic Institute, Florida Atlantic University, is going to be your guide for the next hour.
Hello everyone. Like Shirley said, my name is Megan Conkling. I'm a research scientist at Florida Atlantic University in the United States, and I'm on this research cruise to cryopreserve the benthic marine invertebrates that we collect, as well as train our collaborators in order to develop their own living biobank here in Trinidad. But today's mission on this dive is to look for bubbles and methane seeps. So keep an eye out. We have an octocoral at the bottom. as well as a black coral. Another black coral. Looks like some more zoanthids with a squat lobster, and we have an anemone as well. Small fish. So it looks like we're at the peak of the ridge here. We have a sea star. Some open mussel shells there. coral of some kind with a squat lobster in it. Another halosaur fish.
Yeah, let's agree and then go to below to see if we have some sediment, then we want to make the
Yeah,
the sediment.
Okay.
So this will work great.
All right. Looks like we have an urchin at the bottom. Looks like we have a benthic ctenophores, maybe a crinoid, and some corals. Looks like a squat lobster there. Octocoral, brittle stars.
Hey, I have a data waypoint for you. Thank you very much.
So we're now on our way down to waypoint 4, which is at the base of this ridge. So we're going to be slowly moving our way down. And once we get there, if the environment is right, we'll hopefully be making some heat probe measurements. a coral with a bunch of brittle stars on it.